Find a comfortable position. Dim the lights if you can. Tonight, we are going somewhere that exists right now on this planet, that most people will never see, and that science has barely begun to describe. The title of this documentary uses a provocative phrase, "Science refuses to name." That is not quite accurate, and the true situation is actually stranger. Science is not refusing. It is working carefully on a backlog so large that it would take every marine biologist currently alive working full-time for decades to clear it. The ocean keeps adding to that backlog faster than anyone can process it. The unnamed things are not unnamed because they are hidden from us. In many cases, they are sitting in jars of preservative fluid in the collections of universities and natural history museums, collected decades ago, waiting for a specialist with the right expertise and the time to look at them properly. That is where we are beginning tonight. Not with monsters, not with mythology, with a very specific and very real problem of scale. If you have found something worth staying for in this quiet kind of exploration, a like and a subscription help more than you might think. They let this continue. In the year 2000, an international scientific consortium launched the most ambitious survey of ocean life ever attempted. They called it the Census of Marine Life. Over the following 10 years, more than 2,700 scientists from 80 countries participated. They deployed ships, submersibles, trawls, acoustic sensors, and underwater cameras across every major ocean basin on the planet. They sampled water, sediment, midwater columns, coral reefs, hydrothermal vents, polar seas, and open ocean. The effort cost hundreds of millions of dollars and consumed roughly 650 ship days of survey work per year for an entire decade. In 2010, they published their results. The census identified approximately 250,000 known marine species and estimated that somewhere between 500,000 and 700,000 more species remain to be discovered, described, and named. Some estimates pushed that number considerably higher. The ocean, after 10 years of the most systematic effort in the history of marine science, was still more unknown than known. But those numbers, as striking as they are, understated the problem in one critical way. The census was not evenly distributed. The zones that were sampled most thoroughly were the ones that were easiest to reach: shallow coastal waters, tropical reefs, the upper few hundred meters of the open ocean. The zones that were sampled least thoroughly were the ones that were hardest to reach: the deep midwater, the abyssal floor, the Hadal trenches. And those least sampled zones happen to be, by volume, the majority of the ocean. Consider the numbers. The average depth of the ocean is about 3,700 meters. The sunlit zone, where photosynthesis is possible and where most of the ocean life that anyone has described in any detail actually lives, extends from the surface to roughly 200 meters. That is less than 6% of the total depth. Everything below that is dark. At least 94% of the ocean's volume by depth lies in permanent darkness. And the organisms that live in that dark have been studied, collectively, for a fraction of the time and resources devoted to the sunlit fraction above them. So when we talk about what science refuses to name, what we are really talking about is this: a vast, dark, pressurized volume of water that we have physically visited in scattered locations, under difficult and expensive conditions, for a very short period of time, and in which we keep finding things that do not match anything in our existing records. Things get named when a researcher with the relevant expertise has the time and the preserved specimens and the funding to write a formal taxonomic description. The pipeline between discovery and naming is slow. The ocean keeps generating discoveries faster than the pipeline can process them. What follows tonight is an account of what we know is in that dark. The things with names, the things with tentative identifications, the things detected by chemistry and genetics but never physically seen, and the things that were seen once, filmed once, from an ROV in a location far from any land, and never seen again. Begin with the mesopelagic zone. It extends from 200 meters to roughly 1,000 meters below the surface, below the last usable sunlight, above the true abyssal dark. The water there is dim at the top and completely black at the bottom. The temperature drops sharply from the warm surface layer. Pressure increases from 20 atmospheres at 200 meters to 100 atmospheres at 1,000 meters. 100 times the pressure you are under right now. For most of the 20th century, oceanographers thought of the mesopelagic zone as a transition, something between the productive sunlit zone above and the sparse, cold, deep zone below, important but thin on life. The estimates of fish biomass in this zone, which were based primarily on acoustic surveys and trawl samples, suggested something in the range of 1 billion tons of fish globally, spread across all the world's oceans. In 2014, a paper published in the journal Nature Geoscience revised that estimate. A team of researchers analyzed data from a global acoustic survey that had measured the intensity of sound scattering layers in the mesopelagic zone, which are the echoes produced by the dense concentrations of organisms moving together. The acoustic methods had previously been known to underestimate fish biomass because the sound frequencies used did not reflect well off certain fish species. The 2014 team corrected for this and arrived at a new figure. The mesopelagic zone, they concluded, likely contains somewhere between 1 billion and 10 billion tons of fish, 10 times higher than the previous estimate, possibly 10 times higher than the entire current global commercial fish catch, which is itself around 80 million tons per year, taken every year at significant effort. 10 billion tons of fish living in the darkness between 200 and 1,000 meters, most of them unnamed or poorly described, all of them part of an ecosystem that we cannot see from the surface and can barely reach even when we try. To understand why those fish are there, you need to understand one of the most remarkable mass movements of life on this planet. It happens every day. It has been happening every day for tens of millions of years. It involves more individual organisms than any other animal migration in existence, and almost no one outside of marine science is aware of it. It is called DL vertical migration. At dusk every day across every ocean on Earth, an enormous quantity of marine organisms rises from the mesopelagic zone toward the surface. They travel upward through hundreds of meters of water, moving against the pressure gradient, following the fading light, arriving at the surface layer after dark. There, they feed. On zooplankton, on phytoplankton, on smaller organisms, on each other. The surface at night in the open ocean is a feeding zone for billions of creatures that are invisible during the day because they are hundreds of meters below it. At dawn, the migration reverses. The organisms descend again, back into the mesopelagic zone, back into the dark, where they spend the daylight hours resting, digesting, and avoiding the predators that hunt by sight in the illuminated surface water above them. This migration is so massive and so consistent that it produces a detectable echo on acoustic instruments. The dense layer of organisms moving up and down creates what oceanographers call the deep scattering layer, a broad band of biological material that reflects sound like a false seafloor. The deep scattering layer was first noticed during World War II, when naval sonar operators detected what appeared to be a second seabed that rose toward the surface at night and descended during the day. For a brief period, there was genuine uncertainty about what was causing it. The answer, when it came, was biological. Billions of organisms migrating in coordinated mass every 24 hours. The organisms responsible for this migration are diverse. Many are small fish. Of those, the lanternfish are the most numerous and the most consequential. Lanternfish belonging to the family Myctophiidae are small, typically between 2 and 15 centimeters long, and almost universally unremarkable in appearance. They are dark-bodied, with large eyes adapted to the low light of the mesopelagic zone, and along their sides and bellies they carry rows of small bioluminescent organs called photophores, which produce cold light. The pattern of these photophores varies by species, and is one of the primary means by which species are distinguished from each other. There are approximately 250 described species of lanternfish. They are found in every ocean, at every longitude, from polar seas to the tropics. In terms of sheer numbers of individual fish, lanternfish are estimated to be among the most abundant vertebrates on the planet. In terms of total biomass, they may account for a significant fraction of all the mesopelagic fish biomass that the 2014 study identified, and yet the majority of what is known about individual lanternfish species is limited to their external morphology, their geographic distribution, and their basic diet. The reproductive biology of most species is poorly understood. The specific details of their migration, which populations move how far and on what triggers, vary significantly between species and between ocean regions, and are known in detail for only a small number of the 250 described species. The role they play in the global carbon cycle. carrying carbon from the surface zone down into the deep ocean through their bodies and excretions during the daily migration, has been estimated but not precisely measured. In 2020, a study in the journal ICES Journal of Marine Science attempted to quantify the contribution of mesopelagic fish to carbon transport and concluded that the existing estimates were almost certainly too low, partly because the total biomass of the fish was larger than previously thought, and partly because the behavior of individual species during migration had not been accounted for consistently. The study called for more direct observation of mesopelagic fish in their natural habitat, something that is difficult to accomplish when the habitat in question is 500 meters below the surface in complete darkness. Alongside the lanternfish in the mesopelagic zone are the hatchetfish. These are even stranger in appearance. They belong to two related families and are named for the shape of their body, which is laterally compressed and deepened on the ventral surface, resembling the blade of a small hatchet. Their eyes are tubular, pointing upward, adapted to detect the silhouettes of prey above them against the faint light filtering down from the surface. Their mouths are disproportionately large relative to their body size, and on their undersides, like the lanternfish, they carry photophores. But the hatchetfish photophores serve a specific and remarkable function. They produce downward-pointing light calibrated to match the intensity and wavelength of the faint sunlight filtering down from above. This is called counterillumination. By lighting their own bellies from below, the hatchetfish erase their own silhouette. A predator, looking upward from the darker water below, cannot see the shadow that would normally reveal the fish against the light above. The hatchetfish becomes, in effect, invisible from below. This is not a hypothesis. It has been confirmed in controlled experiments. Hatchetfish adjust the intensity of their ventral photophores in response to changing light conditions, brightening them when the surface light above is brighter and dimming them when it is dimmer. The adjustment is dynamic and continuous, and many hatchetfish species are known only from a handful of specimens. The details of how they calibrate this system in the open ocean, in real conditions, across the full range of light environments they experience during migration, remain subjects of active research. The third major group of mesopelagic fish are the bristlemouths, belonging to the family Gonostomatidae and several related groups. Bristlemouths are believed by some ichthyologists to be the most numerous vertebrates on Earth in terms of individual animals, exceeding even the lanternfish. They are small, typically only a few centimeters long, and their name comes from the tiny sharp teeth that line their jaws. They are not well known to most people, and have not become cultural symbols in the way that anglerfish or sharks have. They are simply very numerous, living in enormous quantities in the mesopelagic zone across all oceans, playing a substantial role in the food web of the deep sea and studied in comparative depth by only a small number of specialists. The taxonomy of bristlemouths is genuinely unresolved in places. The relationships between some genera and species are disputed. Cryptic species, meaning populations that look alike but are genetically distinct enough to be considered separate species, have been identified in several groups using molecular techniques in recent years. The true number of bristlemouth species globally is not known with confidence. All of these fish, the lanternfish and the hatchetfish and the bristlemouths, are participants in a daily migration that moves a staggering quantity of biological material between the surface ocean and the deep ocean. They eat at the surface, and they carry the carbon from that eating down into the deep. When they excrete, or when they die and sink, that carbon becomes part of the deep ocean carbon reservoir rather than returning to the atmosphere. The mesopelagic zone is, in this sense, a pump, a biological mechanism that draws carbon from the surface and deposits it in the deep. The efficiency of that pump depends on the total biomass of the organisms in the zone, on the distance they migrate, on how much carbon they sequester versus how much they respire at depth, and on the community composition of the zone, meaning which species are present in what proportions. All of those variables are, to varying degrees, imprecisely known. The 2014 revision of the biomass estimate changed the calculations for the biological carbon pump substantially. How many more revisions are still coming as better measurements are made of the organisms that make it work is a question that researchers working in this field cannot yet answer. So, beneath 200 meters of water in every ocean on Earth, there are somewhere between 1 and 10 billion tons of fish. Most of them make a daily journey of hundreds of meters. Most of them belong to families and genera that are described, but most individual species within those families are known in detail only from dead specimens. The living behavior, the real-time ecology, the precise role in the carbon cycle, that is where the science becomes sparse. And the mesopelagic zone is the best understood of the deep zones. Below 1,000 meters, things become considerably more uncertain. To appreciate why the mesopelagic zone remained so poorly understood for so long, it helps to think about what it actually takes to study it. You cannot dive there with conventional scuba equipment, which limits researchers to roughly 40 meters. You cannot observe it from the surface. You cannot see into it. The acoustic surveys that detected the deep scattering layer gave researchers a sense of the density and movement of organisms, but acoustic returns do not tell you which species you are looking at, how large the individual animals are, or what they are doing. To collect specimens, researchers have used trawl nets pulled through the water column at depth. But trawl nets collect organisms damaged and stressed, pulled through every depth zone on the way back to the surface. Organisms that live at 600 meters are dragged through the 300-meter zone, the 100-meter zone, and finally the surface, with dramatic changes in pressure and temperature throughout. Delicate organisms are destroyed. Soft-bodied organisms lose their structure entirely. What arrives on deck is often a compressed, damaged mass of material from which species identification is difficult, and behavioral inference is impossible. Researchers have therefore turned to optical methods. Cameras on remotely operated vehicles lowered into the mesopelagic zone to observe organisms in their natural habitat. This approach has yielded remarkable footage, but it is expensive, limited to specific geographic locations, and sensitive to season, time of day, and local oceanographic conditions. A single ROV dive covers a tiny volume of water. The mesopelagic zone covers the entire global ocean. The result is that even for the most numerically abundant vertebrates on Earth, we know primarily what they look like and roughly where they are. Their behavior, their breeding, their interactions with each other and with other organisms in the zone, the specific mechanisms by which they navigate their daily migration, the sensory cues they use to know when to ascend and when to descend, these remain areas of active investigation with substantial unknowns. And that is for the described species, the ones with names. Within the mesopelagic zone, there are also organisms that have not been formally described at all. Environmental DNA surveys of mesopelagic water, which sequence the genetic material shed by organisms into the water column without requiring physical collection of those organisms, consistently detect genetic signatures that do not match any species in existing databases. These are called dark taxa. Genetically real, ecologically present, taxonomically invisible. They are there. We cannot say what they are. The gelatinous zooplankton of the mesopelagic face the same problem. The salps, the pyrosomes, the medusae. These are organisms with soft, fragile bodies that trawl nets destroy and that cameras capture only when the ROV happens to be in the right location at the right moment. Salps in particular can form colonies tens of meters long, moving through the water in coordinated pulsing waves, and are among the more dramatic residents of the mesopelagic zone. Their role in packaging and sinking organic material out of the surface zone is significant. How significant precisely remains poorly constrained. Now think about what all of this means for the title of this documentary. What is really living in the deep ocean that science has not yet named? In the mesopelagic zone alone, the answer involves bristlemouth fish species whose taxonomy is contested, lanternfish species known from single specimens collected decades ago, hatchetfish species detected by environmental DNA but never physically observed, gelatinous organisms too fragile for conventional collection, and entire ecological interactions that have never been directly witnessed. The mesopelagic zone is not even the deep ocean by most definitions. It begins at 200 meters, and already the unknowns are substantial. Below the mesopelagic zone, the water gets darker and the unknowns get larger. The bathypelagic zone extends from 1,000 meters to 4,000 meters. Below that is the abyssopelagic, from 4,000 to 6,000 meters. And below that, in the trenches, is the hadal zone, reaching nearly 11,000 meters at its deepest point. As depth increases, pressure increases, temperature decreases, food becomes scarcer, and the organisms that have evolved to survive there become progressively more alien to surface biology and progressively less known to science. The bathypelagic zone is where you find the anglerfish, the dragonfish, the fang-tooth. It is where you find the giant squid. It is where the sounds detected by hydrophones grow stranger and less attributable to known sources. It is a zone visited by cameras far less often than the mesopelagic, and sampled by trawls even less reliably, because the organisms there are slower, more delicate, and more specialized for conditions that conventional sampling methods destroy. Below the bathypelagic, the abyssopelagic zone covers the vast open water above the ocean floor in the deepest basins. And then there is the floor itself. The abyssal plain occupies the majority of the ocean floor. It is a vast, cold, nearly featureless expanse of fine sediment extending for thousands of kilometers in every direction, lying between 3,000 and 6,000 meters below the surface. The pressure there is between 300 and 600 times the pressure at sea level. The temperature is typically between 1 and 4 degrees Celsius, just above freezing. There is no light of any kind except what is produced by the organisms themselves. The food supply is thin. Particles of organic matter that fall from the productive zones above. A rain of microscopic material called marine snow, arriving at the floor after a journey of weeks or months through the water column above. In this environment, life exists. It is not sparse in terms of species. It is sparse in terms of the density of individual organisms per square meter, because the food supply will only support a limited biomass. But across the vast area of the abyssal plain, that sparse life adds up to something substantial, and much of it is either poorly described or not described at all. Among the strangest inhabitants of the abyssal floor are the xenophyophores. These are single-celled organisms. One cell. They have no brain, no nervous system, no division of labor between different cell types, because there are no different cell types. They are a single unit of biological activity contained within one plasma membrane. And yet the largest species, Syringamina fragilissima, described from the Atlantic Ocean, can reach 20 centimeters in diameter. 20 centimeters, the size of a dinner plate, and it is a single cell. To grow to this size, xenophyophores do something unusual even by the standards of strange cellular biology. They do not divide. Instead, the nucleus of the cell replicates repeatedly, producing thousands of nuclei that remain within the same cytoplasm, sharing the same membrane, functioning as one. The organism builds itself a structure, called a test, from sediment particles and organic debris glued together with its own secretions. This test can be fragile and intricate, looking something like a loosely organized sponge or a mass of tubes. The xenophyophore lives inside this self-constructed architecture on the seafloor, extending its cytoplasm through the test to feed on the organic particles in the sediment around it. Xenophyophores have been found at remarkable depths. Specimens have been collected from more than 10,000 meters, deeper than most multicellular organisms survive. They appear to be among the most pressure-tolerant eukaryotes known to science. Their biology at these extreme depths, how they maintain cellular function under pressures that would destroy the proteins of most organisms, is not well understood. There are approximately 50 described species of xenophyophore. Researchers who specialize in the group believe the actual number of species is considerably higher. Because they are fragile, bottom-dwelling, and live in places that are difficult to visit, collection is difficult, and specimens are often damaged beyond recognition. When ROVs have photographed the abyssal floor in areas not previously surveyed, they have frequently encountered xenophyophores that cannot be confidently assigned to any described species. Moving across the same sediment as the xenophyophores are the holothurians, the sea cucumbers. These are not the small, slug-like animals of tidal pools and coral reefs. The deep-sea holothurians are different organisms adapted to different conditions. Among the most abundant are the species of the genus Scotoplanes, commonly called sea pigs. They are pale, translucent, roughly the size of a large grapefruit, with short, stubby legs used to walk along the sediment surface and tube feet used to gather food. They are found in enormous numbers across the abyssal plains of the Pacific and Atlantic. In some surveys, they are the most abundant megafaunal organism visible on the seafloor. What sea pigs are doing in such quantities on the abyssal plain is reasonably well understood at the population level. They are consuming organic material in the sediment, processing it, and excreting it. They are, in a sense, the earthworms of the deep sea floor, aerating the sediment and cycling nutrients through their bodies as they move. But the specific ecology of individual species, how long they live, how they reproduce, what their specific food preferences are, how their populations respond to changes in the organic input from above, is poorly documented for most species. There are over 1,700 described species of holothurian globally. How many deep-sea holothurian species remain undescribed is not known. Trawl samples from the abyssal plain consistently include individuals that cannot be confidently assigned to any described species. The group is large enough and the deep-sea fauna is poorly enough sampled that new species are described regularly, and the end of that process is not in sight. Less visible than the sea pigs, but arguably more remarkable, are the bone-eating worms of the genus Osedax. These were discovered in 2002, when researchers studying a gray whale carcass placed on the floor of Monterey Canyon at about 2,900 meters depth found the bones covered with feathery, flower-like structures attached to the bone surface. The structures were female Osedax worms. They had no mouth, no gut, no stomach. They had penetrated the bone with root-like extensions of their tissue, and within those roots they hosted symbiotic bacteria that broke down the fats and proteins in the bone, providing the worm with nutrients. The males were microscopic, living in a gelatinous tube on the female's body, reduced to essentially reproductive tissue with little else. A large female could host dozens of males. The worms were a complete biological surprise. They represented a new genus, a new species, and a newly understood ecological role, the cleanup of whale carcasses on the deep seafloor, a resource type that had not previously been studied as a habitat. Since the discovery of Osedax in Monterey Canyon, more than 20 species have been described from different ocean basins. Phylogenetic analysis shows that the genus is older than the whales it primarily feeds on, suggesting that Osedax evolved on the bones of marine reptiles or marine mammals that preceded whales in the deep ocean fossil record, and then transferred its ecological role to whale carcasses as those became abundant. The diversity of Osedax species is almost certainly higher than the 20 or so currently described, because the deep ocean contains many more whale fall sites than have been visited and studied. Now consider what happens acoustically in the part of the ocean occupied by all of these organisms. The ocean transmits sound with extraordinary efficiency. In the deep ocean, a specific combination of pressure, temperature, and salinity creates what oceanographers call the SOFAR channel, the sound fixing and ranging channel. It is a horizontal layer of water, typically centered between 600 and 1,200 meters depth, in which the speed of sound reaches a local minimum. Because sound naturally bends toward the layer of minimum speed, sounds produced within the SOFAR channel tend to be trapped there, traveling horizontally for vast distances with very little loss of energy. A whale calling in the North Atlantic can be detected by hydrophones in the North Pacific. The sound has traveled across an ocean basin while staying within the channel. A single explosion underwater can be detected thousands of kilometers away. The SOFAR channel is, effectively, a global acoustic network, and anything that produces sound within it is broadcasting that sound across an enormous area. During the Cold War, the United States Navy installed a network of hydrophones throughout the world's oceans called SOSUS, the Sound Surveillance System, designed to detect Soviet submarines. After the Cold War, portions of this network were made available to civilian researchers. For the first time, marine scientists had access to long-term continuous acoustic recordings from the deep ocean. What they heard was unexpected. The ocean, they discovered, is not quiet. It is full of sounds, most of them identifiable. The grinding of tectonic plates, the rumble of distant earthquakes, the calls of whales, the noise of shipping traffic. But scattered among these identifiable sounds were others that did not fit any known pattern. In 1997, NOAA's hydrophone arrays detected a sound that became known as the bloop. It was an ultra-low-frequency, high-amplitude sound that rose in frequency over roughly 1 minute. It was so powerful that it was detected by instruments more than 5,000 kilometers apart. For several years, its source was genuinely unknown. The frequency profile and the intensity both suggested a very large biological source, possibly an animal far larger than any known living species. That speculation generated considerable public attention. The eventual scientific consensus was less dramatic, but no less interesting. Analysis of the bloop's sound characteristics matched the acoustic signature produced by ice quakes, the cracking and calving of large ice sheets in Antarctica. The sound had traveled thousands of kilometers through the SOFAR channel from its Antarctic source to the hydrophone arrays. It was real. It was just not biological. But the Mariana Trench has produced its own anomalous sounds that have not been resolved as quickly. In 2014 and 2015, researchers deploying deep-sea acoustic recorders in the Mariana Trench recorded a sound they named the Western Pacific Biotwang. It was a complex, five-part call lasting about 3.5 seconds. It began with deep moaning tones, shifted through several frequency changes, and ended with a high-pitched metallic sound unlike anything in the catalog of known marine mammal calls. The leading hypothesis is that it is a minke whale call of a type not previously documented from the Pacific. But no one has ever directly observed a minke whale making this call, and its unusual mechanical quality does not resemble any other known minke whale vocalizations. The NOAA hydrophone arrays have also detected several other anomalous sounds over the decades. Slowdown is a sound that decreases in frequency over about 7 minutes, first detected in 1997 near the equator in the Pacific. Train is a repetitive, regular sound detected in 1997. Upsweep is a signal that increases in frequency over time, detected seasonally from a location near the southern Pacific. These sounds have in common that they are not definitively attributed to any known biological or geological source. The seasonal periodicity of upsweep suggests something with a biological or oceanic cycle driving it, but what that something is remains unknown. And then there is the sound known as the 52 Hz whale. In 1989, a United States Navy hydrophone array in the Pacific detected a call at 52 Hz. This is unusual because blue whale calls, which are the lowest frequency calls of any whale, typically occur between 10 and 39 Hz. Fin whale calls are typically around 20 Hz. A call at 52 Hz is significantly higher than either species normally produces, and it does not match the pattern of any other known whale species. The call was detected repeatedly over subsequent years, always alone, always traveling at speeds consistent with a large whale, always at the same unusual frequency. Because it was never accompanied by any other whale calls of the same frequency, the working hypothesis among marine mammal researchers has been that this is a single individual whale, possibly a hybrid or an individual with a physical abnormality, calling at a frequency that no other whale of its kind produces, and therefore receiving no response. Whether the 52 Hz whale is lonely in any emotional sense is a question biology cannot answer. Whether it is anatomically unusual is also unknown because it has never been visually confirmed. The call has continued to be detected periodically for more than 3 decades. The animal that makes it has never been seen. The sounds that the SOFAR channel carries, the ones we can name and the ones we cannot, are produced by organisms that live in a zone scientists have long considered the most inaccessible part of the ocean, not the trenches, not the abyssal floor. The midwater column itself, the water between the surface and the floor in the deepest parts of the ocean, is not crossed by anything traveling from surface to seafloor. Most objects that sink slowly. ROVs descend through it vertically, quickly, their cameras not designed to survey it as they pass. The midwater column of the deep ocean is, in many ways, the least understood physical space on the planet. It is in this space that one of the more unsettling deep-sea observations was made. In 1988, a Japanese research submarine operating near the Ogasawara Islands filmed something the crew could not immediately identify. The footage showed a large squid with extremely elongated, thin arms extending from the body at an unusual angle. The arms appeared to be held horizontally away from the body, with tentacles hanging straight down from the elbows of a right-angle bend. The overall appearance was unlike any squid known at the time. Over the following years, similar animals were filmed by other remotely operated vehicles in the deep Atlantic, Pacific, and Indian Oceans, almost always in the bathypelagic zone between 1,000 and 4,000 meters. The squid became known informally as the bigfin squid, named for the proportionally large fins relative to its body, or as magna pinna, meaning big fin in Latin, which became the name of the genus when formal descriptions were eventually written. The formal description of magna pinna was complicated by the fact that no adult specimen had ever been physically collected. The animals filmed were juveniles or subadults. Their arms, in the filmed individuals, were sometimes estimated at lengths between 4 and 8 meters, suggesting that adult animals, with proportionally longer arms, might reach sizes that would make them among the largest squid in the ocean. But without a collected adult, the size range of the adult animal remains unknown. The diet is unknown. The reproductive behavior is unknown. The lifespan is unknown. The total population size is unknown. What is known is that magna pinna appears to be geographically widespread, occurring across multiple ocean basins, and that it inhabits depth ranges that are among the least visited by research equipment. It was probably not rare before it was discovered. It was simply in a place where cameras had not been pointed. How many animals like this exist in the deep mid-water, present and ecologically significant but unknown to science because no one has had the equipment or the funding to look where they are, is a question without a reliable answer. In 2004, MBARI, the Monterey Bay Aquarium Research Institute, was conducting a deep survey in Monterey Canyon when their ROV Tiburon encountered, at approximately 2,400 meters depth, a red jellyfish with a bell nearly 1 meter in diameter. Its oral arms were extended horizontally in an unusual posture, with tentacles hanging below them in long curtains. The research team on the surface could not immediately identify it. It was eventually identified as Stygiomedusa gigantea, one of the largest jellyfish in the ocean. Known from fewer than 20 observations in all of recorded deep-sea research, despite appearing to be a successful and widespread species. Stygiomedusa had been described from damaged specimens brought to the surface in trawl nets since the mid-20th century. In those damaged specimens, the shape, the posture, and the full scale of the living animal were not apparent. The 2004 footage was among the first extended records of a living specimen in its natural habitat. It showed an animal behaving in a way that was not predictable from the dead specimens. The horizontal arm posture appeared to be a passive feeding strategy, holding the arms out to intercept small organisms swimming through the water column. That interpretation had not been possible from dead material. This is a recurring pattern in deep-sea biology. The living animal and the collected specimen are often so different in appearance and posture that researchers working only with collections cannot accurately visualize what the living animal looks like or how it behaves. Deepsteria, a genus of large, thin, membraned jellyfish that can expand their bells to nearly a meter across, was described in 1967 from a single damaged specimen. When ROV footage finally captured a living Deepsteria in its natural habitat, researchers on the surface initially believed they were looking at an unknown organism. The living animal, fully expanded, was unrecognizable as the same species as the preserved specimen in the collection. The gap between the described organism and the living one is one of the fundamental challenges of deep-sea taxonomy, and it is a gap that can only be closed by more direct observation of organisms in their natural habitat. That observation requires equipment and ship time, which require funding, which is allocated by institutions and funding agencies that must weigh the costs of deep-sea research against other priorities. The hydrothermal vents represent a different kind of unknown. Not a matter of organisms present but not seen, but of entire ecosystems whose existence was not suspected until the late 20th century. In February 1977, a team of scientists aboard the research submarine Alvin descended to the Galapagos Rift, a spreading center on the floor of the Pacific Ocean, to investigate temperature anomalies detected by previous oceanographic surveys. The anomalies suggested that unusually warm water was rising from the seafloor in a specific location. At approximately 2,500 meters depth, Alvin's lights illuminated what the researchers later described as something none of them had expected to see. A dense community of organisms clustered around vents releasing warm, chemical-rich water from the seafloor. The organisms around the vents were not the sparse deep-sea fauna that had been the standard expectation for the deep ocean floor. They were dense. They were large. There were tube worms with blood-red plumes, over 2 meters long, emerging from white tubes attached to the basalt. There were clams the size of dinner plates. There were crabs and shrimp moving through the community. There were white mats of bacteria covering the rocks. None of these organisms were powered by photosynthesis. The sunlight that drives nearly all surface life cannot reach 2,500 meters. These organisms were living on chemical energy, the hydrogen sulfide and other reduced compounds flowing from the vents, which bacteria oxidize to produce energy in a process called chemosynthesis. The bacteria formed the base of the food web, just as phytoplankton form the base of the surface food web. But the energy source was not light. It was chemistry from the Earth's interior. The discovery was revolutionary. It demonstrated that life could exist and thrive in complete darkness, under conditions of extreme pressure and temperature, without any dependence on the sun. It was the first clear demonstration that the sun is not a prerequisite for life. That finding has since been extended in imagination to the possibility of life in other places in the solar system, in the subsurface oceans of Europa, Enceladus, and other moons. where liquid water in contact with a rocky seafloor might produce the same conditions that vent ecosystems exploit on Earth. The tube worms at the Galapagos Rift were eventually named Riftia pachyptila. They are among the most studied deep-sea organisms in existence, largely because they are dramatic, accessible by ROV, and present at multiple vent fields visited regularly. But even for Riftia, some aspects of the biology are only partially understood. The mechanism by which the worm regulates the delivery of sulfide to its internal bacterial symbionts while simultaneously preventing the sulfide from poisoning its own tissue is a biochemical system that has been studied in detail, but that still contains unanswered questions about the molecular specifics of the regulation. And Riftia is the well-studied exception. Most vent species are not Riftia. Vent communities globally contain hundreds of described species, many of them known from only one or a small number of vent fields. The total number of hydrothermal vent fields on the ocean floor is not precisely known. Estimates based on the length of mid-ocean ridges and the frequency with which active vents have been found along surveyed sections suggest that thousands of active vent fields exist globally. The number that have been biologically surveyed in any detail is in the low hundreds. Each new vent field surveyed for the first time has, consistently, produced species not found at other vent fields. Cold seeps are a related but distinct phenomenon. Where hydrothermal vents are associated with tectonic spreading centers and the outflow of geothermally heated water, cold seeps occur where methane or hydrogen sulfide migrates upward through the sediment from underlying geological formations. The fluid that reaches the seafloor at a cold seep is cold, not hot, but it is chemically rich, and it supports chemosynthetic communities similar in structure to vent communities. Bacterial mats, tube worms, mussels with symbiotic bacteria in their tissue, clams, crabs, and fish that exploit the productivity of the chemosynthetic base. Cold seeps are more geographically widespread than hydrothermal vents, occurring along continental margins and in other locations where the appropriate geology exists. The number of cold seep sites globally is also not precisely known. They are difficult to detect from the surface without targeted acoustic surveys looking for the methane bubbles that often emerge from active seeps. New cold seep sites are discovered regularly, and each one brings its own community of organisms, some described and some not. The question of gigantism in the deep sea deserves its own examination because it runs counter to the intuition that extreme environments produce small, simplified life forms. The opposite is often true in the deep ocean. Where surface relatives are small, deep-sea relatives are sometimes dramatically larger. This pattern, called deep-sea gigantism, is well documented but not fully explained. The giant isopod is the most recognizable example. Isopods are crustaceans related to the common pillbug or roly-poly found in garden soil. Surface isopods are typically less than a centimeter long. The giant isopod, Bathynomus giganteus, can reach 76 centimeters and weigh over 1.7 kilograms. It has a segmented armored body, compound eyes, 7 pairs of legs, and lives on the continental margins and abyssal plains of the Atlantic, Pacific, and Indian Oceans. It is a scavenger, feeding primarily on dead fish, whale falls, and other organic material that sinks to the seafloor. It can survive for years between meals, slowing its metabolism to a degree that surface crustaceans cannot achieve. Why is it so much larger than its surface relatives? Several hypotheses have been proposed. Cold temperatures slow metabolic rates and may extend the period of growth before reproduction, leading to larger body size at maturity. The episodic feast and famine nature of food supply on the abyssal floor may favor larger body size because larger animals can store more energy and survive longer between meals. The absence of certain predators that constrain size in shallow environments may allow deep-sea animals to grow to sizes that would be ecologically unsustainable near the surface. None of these hypotheses is fully satisfactory on its own. The true explanation for deep-sea gigantism is likely a combination of factors, and it probably varies between taxonomic groups. What is clear is that the pattern is real. Giant versions of surface animals adapted to the cold, dark, pressurized environment of the deep ocean, living in ways that we can describe in outline but not always in detail. The colossal squid, Mesinicoteuthis hamiltoni, is the largest invertebrate on Earth. The largest confirmed specimen, a female caught in 2007 in Antarctic waters, measured approximately 10 meters in total length and weighed around 495 kilograms. Its eyes, at up to 30 centimeters in diameter, are the largest of any known living animal. They are thought to be adapted to detecting the faint bioluminescent flashes produced by sperm whales as those predators hunt in the dark Antarctic depths. The colossal squid has rotating hooks on its tentacles rather than the suckers of most squid species. These hooks can swivel independently, giving the tentacles a gripping capability suited for capturing large, struggling prey. Its prey is thought to be primarily Patagonian toothfish and other large deep-water fish. Its predators are primarily sperm whales, which have been found with colossal squid beaks in their stomachs and with circular scars on their skin that match the diameter of colossal squid tentacle hooks. Despite this indirect evidence that colossal squid are prey for sperm whales in significant quantities, no living colossal squid has ever been observed in its natural habitat, not once. The animal exists, in living form, in a depth range and geographic location that ROVs have visited only a small number of times. What we know of it comes entirely from carcasses, from stomach contents of predators, from fragments recovered in fishing gear, and from the physical examination of the rare specimens that have been collected in reasonable condition. How it hunts, how it reproduces, how it navigates the Antarctic deep, what its lifespan is, how it interacts with other organisms in the community it inhabits, all of this is unknown. The anglerfish of the deep ocean present a different kind of biological surprise. There are over 200 described species of anglerfish. The female anglerfish is the recognizable form, a large-jawed, fang-toothed fish with a modified dorsal spine, the illysium, ending in a bioluminescent lure called the esca, which hangs in front of the fish's mouth. The esca produces light through a symbiotic relationship with bioluminescent bacteria housed within it. The female uses the lure to attract prey in the complete darkness of the deep ocean, and her mouth and stomach are designed to accommodate prey nearly as large as herself. The males of most deep-sea anglerfish species are dramatically smaller, often less than a centimeter long, and cannot feed independently as adults. When a male anglerfish locates a female, which he does using his highly developed olfactory system to detect her pheromones, he bites into her skin. His jaw and her skin begin to fuse. Over weeks, the tissues merge. The male's own body begins to atrophy. His eyes, his fins, most of his internal organs. His circulatory system connects to hers. He receives nutrients through her blood. In return, he provides sperm when she is ready to reproduce. A single female may carry multiple males attached to her at once, each one fully incorporated into her body. This process, called sexual parasitism, is unique among vertebrates. It was first described correctly in the 1920s, and the mechanism by which the female's immune system suppresses its own rejection of what is effectively foreign tissue, something that would be catastrophic in most vertebrates, is only partially understood at the molecular level. The immune suppression appears to involve modifications to both the female's immune response and the male's surface chemistry, but the precise mechanism has not been fully worked out for most of the 200-plus anglerfish species. The dragonfish of the family Stomidae represent yet another unusual deep-sea adaptation. These are elongated, dark-bodied fish with disproportionately large jaws and teeth, a chin barbel often tipped with a bioluminescent lure, and rows of photophores along their bodies. What makes certain dragonfish species particularly notable is a visual and photonic capability shared by almost no other organisms in the ocean: the ability to produce and detect red light. In the deep ocean, where water absorbs red wavelengths within the first few meters of the surface, red light effectively does not exist as a natural illuminant. Most deep-sea animals have evolved visual systems sensitive to blue and blue-green wavelengths, because those are the wavelengths present in their environment. Red light cannot be seen by the majority of deep-sea organisms. Several species of dragonfish, particularly in the genera Malacosteus, Aristostomias, and Pachystomias, produce red bioluminescence from suborbital photophores and possess photoreceptors capable of detecting it. This gives them access to a channel of illumination and detection that is effectively invisible to their prey and their competitors. A dragonfish can illuminate the water in front of it with red light and see the organisms illuminated there, while those organisms remain unaware that they are being observed. The biochemical origin of this system is one of the more fascinating problems. In deep-sea sensory biology, animals do not synthesize the chlorophyll-derived pigments that the dragonfish appear to use in their red detection system. The current hypothesis is that the dragonfish acquire these pigments dietarily, through a food chain that ultimately traces back to photosynthetic material consumed near the surface and transferred down through multiple trophic levels. If that is correct, then the dragonfish's private red searchlight is, in some sense, made of recycled sunlight. The siphonophores of the deep ocean represent perhaps the most structurally unusual organisms in the sea. Siphonophores are colonial organisms, composed of many individual animals called zooids, each specialized for a different function, some for swimming, some for feeding, some for reproduction, some for flotation. All zooids in a colony are genetically identical, having budded from the same founding individual. The colony functions as a single organism with different specialized parts, even though it is simultaneously many individual animals. Praia dubia, the longest known siphonophore, can reach lengths of 40 to 50 meters. This makes it one of the longest animals on Earth, certainly the longest colonial animal. Its body is composed of swimming bells at the front, which power the colony's movement through rhythmic pulsing, and a long stem behind, from which hang the feeding and reproductive zooids. The feeding zooids extend tentacles armed with stinging cells, creating a curtain of capture mechanisms trailing behind the swimming bells. Siphonophores are extremely fragile. Conventional trawl nets destroy them. Most of what is known about their living form and behavior comes from in situ observations by ROV cameras, which have revealed that their behavior in the water is very different from what the damaged specimens in collections suggested. The total number of siphonophore species is estimated at around 175 described, but researchers who specialize in the group believe the actual diversity is higher, with many bathypelagic and abyssopelagic species remaining to be described. Bioluminescence is the thread connecting most of the organisms we have discussed in the deep ocean. It is not an occasional curiosity here. Below 200 meters, it is the primary mode of light in the environment. Studies of deep-sea organisms have suggested that somewhere between 76 and 90% of all deep-sea species are capable of producing light. This is not a minority adaptation. It is the dominant sensory reality of the deep. The chemistry of bioluminescence involves a class of compounds called luciferins, which react with oxygen in the presence of an enzyme called luciferase to produce light with very little waste heat. Different taxonomic groups have independently evolved different luciferin-luciferase systems. The bioluminescent systems in dinoflagellates, in fish, in jellyfish, in squid, and in shrimp are chemically distinct from each other, meaning that bioluminescence has evolved separately, multiple times, in groups that share no recent common ancestor. The ecological functions of bioluminescence in the deep sea are diverse. It functions as a predatory lure, as the anglerfish's esca demonstrates. It functions as counter-illumination camouflage, as the hatchetfish demonstrates. It functions as a startling defense mechanism. Some deep-sea shrimp squirt clouds of luminescent fluid at predators, temporarily blinding them while the shrimp escapes. And it appears to function in communication, particularly in squid and other cephalopods, where complex patterns of flashing may carry information about reproductive status or individual identity. For most of the species in the deep ocean that possess bioluminescent capability, the specific function of their light production is not known with confidence. The patterns of photophore arrangement on different species have been cataloged. The chemistry of light production has been analyzed in many species. But the behavioral context, what triggers light production, what responses it evokes in other organisms, what ecological outcomes it affects, has been observed directly in only a small fraction of bioluminescent deep-sea species. The deepest environments of the ocean are the Hadal trenches. These are the elongated, narrow depressions in the seafloor created by subduction zones, where one tectonic plate dives beneath another. They extend below 6,000 meters, and in the case of the Mariana Trench reach 10,135 meters at the Challenger Deep, the deepest measured point on the planet's surface. At 11,000 meters, the pressure is approximately 1,100 times the pressure at sea level. This is enough to compress steel. It is enough to change the physical chemistry of water itself, altering its viscosity and the behavior of dissolved substances in ways that surface biology simply does not encounter. Proteins, the molecules that carry out virtually every biological function in living cells, are vulnerable to pressure. High pressure disrupts the three-dimensional shapes that proteins must maintain to function. Organisms living at Hadal depths must have fundamentally different protein chemistry from surface organisms, modified at the molecular level to resist what is called pressure denaturation. How they accomplish this is one of the more active areas of deep-sea biochemistry. The trimethylamine oxide compound, known as TMAO, is found in elevated concentrations in the tissues of hadal fish and appears to counteract the destabilizing effects of pressure on proteins. The deeper a fish lives, the higher its TMAO concentration tends to be. This relationship was documented systematically in a 2017 study that analyzed TMAO concentrations in fish from different depths and found that the correlation was tight enough to predict depth of habitat from tissue chemistry. The trenches are not biologically connected to each other. Each trench is an isolated deep environment, separated from other trench systems by thousands of kilometers of abyssal plain at shallower depths. The organisms that live in the hadal zone of the Mariana Trench are not the same as the organisms in the hadal zone of the Tonga Trench, despite the environmental conditions being broadly similar. This isolation has allowed evolution to proceed independently in each trench system, producing distinct species assemblages even where the ecological roles are similar. The snailfish are a particularly striking example of this. Snailfish, belonging to the family Liparidae, have proven to be the dominant fish predators in the hadal zone across multiple trench systems. They are gelatinous, pale, tadpole-shaped fish that have evolved to function at pressures that would be fatal to virtually all surface fish. In the Mariana Trench, the species Pseudoliparis swirei was formally described in 2017. In 2018, a lander deployed by a team from Newcastle University and the University of Western Australia filmed a snailfish at 8,178 meters depth in the Mariana Trench, at the time the deepest confirmed fish sighting. Subsequent expeditions have recorded individuals at even greater depths. In the Tonga Trench, a different snailfish species appears to dominate. In the Izu Ogasawara Trench, north of the Mariana, there are again different species. The snailfish of each trench system have evolved from surface ancestors that somehow colonized the hadal zone, possibly through the gradual deepening of a population over geological time, and have since diverged from each other through the isolation that the trench structure imposes. The hadal amphipods present the same pattern. Amphipods are small crustaceans, shrimp-like in general appearance, and they are among the most abundant animals in the hadal zone. In any hadal survey, enormous numbers of amphipods are typically attracted to any baited lander within hours of its arrival on the floor. Different trench systems have different dominant amphipod species. The Mariana Trench amphipods are distinct from the Kermadec Trench amphipods, which are distinct from those of the Puerto Rico Trench in the Atlantic. What this means in practice is that the hadal zone, despite being superficially similar from trench to trench, is not a single environment in biological terms. It is a series of isolated biological experiments, each running independently, each producing a distinct evolutionary outcome from the same general set of starting conditions. The total number of hadal species across all trench systems is not known. New trench species are described after virtually every systematic hadal survey. The pace of discovery has not slowed as the surveys have accumulated. In 2019, the 5 Deeps expedition, a privately funded project to reach the deepest point of each of the 5 major ocean basins, conducted multiple dives to the Challenger Deep and other hadal locations. Accompanying these manned dives were autonomous landers that collected biological samples and video footage. The expedition confirmed several new depth records for fish observation and collected specimens of organisms for biological analysis. It also produced footage that the expedition scientists and subsequent specialist reviewers have not been able to fully explain. At one location in the Mariana Trench, a lander camera recorded a translucent, elongated organism drifting slowly through the water column near the floor. It was not a snailfish. It was not any of the amphipod species known from the trench. It moved through the water in a way that suggested both active swimming and passive drifting, with no obvious fin or appendage movement visible in the footage. The footage was distributed to specialists in deep-sea biology. As of the most recent published accounts of the expedition, the organism had not been formally identified. It remains in the category of observations that exist in the scientific literature as unsolved. This category is larger than most people realize, not just for the Mariana Trench. For every systematically surveyed region of the deep ocean, there are observations that did not match anything in the existing databases when they were made, that were logged and passed to specialists, and that remain unresolved because no subsequent observation has produced better footage or a physical specimen. The deep ocean generates these unsolved observations faster than the research community can resolve them. The Greenland shark, Somniosus microcephalus, is one of the few hadal or deep-water organisms whose existence is well known to the general public, largely because of a single extraordinary fact about its lifespan. A 2016 study by a team led by Julius Nielsen at the University of Copenhagen used radiocarbon dating of the crystalline proteins in the eye lenses of Greenland sharks to estimate the ages of individual specimens. Unlike most tissues, which renew themselves throughout an organism's life, the proteins in the eye lens core are laid down during embryonic development and not replaced afterward. They preserve the carbon isotope ratios of the environment at the time of the animal's birth, which can be cross-referenced against the known historical record of atmospheric radiocarbon to estimate age. The oldest individual in the Nielsen study was estimated at 392 years, with an uncertainty range that extended the plausible maximum to approximately 500 years. A Greenland shark 500 years old would have been born before the invention of the telescope, before the settlement of North America by Europeans, before the composition of most of Shakespeare's plays. It would have been alive during the 30 Years' War, during the construction of the Taj Mahal, during the entire period of the scientific revolution. And it might still be alive right now, moving through the cold water of the North Atlantic or Arctic at roughly the pace of a slow human walking, which is approximately its usual swimming speed. The Greenland shark is not a deep trench specialist. It ranges across a broad depth spectrum from relatively shallow water in the Arctic to depths of over 2,000 meters in the North Atlantic and potentially deeper. It is a top predator in its range, consuming seals, fish, squid, and whatever else it can catch or scavenge. Its sensory biology is unusual. The corneas of its eyes are commonly parasitized by a copepod crustacean, Omatocoita elongata, which attaches permanently to the eye and may over time damage the shark's vision. It appears to navigate and hunt primarily by smell rather than sight. Why a shark can live 500 years while most sharks live decades is not fully understood. The cold water metabolism hypothesis is consistent with the data. Cold temperatures slow all biochemical processes, including the accumulation of cellular damage that produces aging. But the quantitative relationship between temperature and lifespan in sharks is not established for most species, and the Greenland shark's longevity is so extreme that temperature alone may not account for it. There may be additional mechanisms, possibly related to DNA repair or the regulation of oxidative stress, that contribute to this unusual longevity. What the Greenland shark demonstrates as a single example from a well-studied taxon in a relatively accessible part of the ocean is the degree to which our understanding of even documented deepwater species is incomplete. If we do not yet fully understand the lifespan mechanisms of one of the most famous large predators in the deep cold ocean, what gaps remain for the hundreds of less studied species that share its habitat? And for the thousands of species in deeper and less accessible zones that we have barely begun to examine? The taxonomy of the deep ocean is not complete. It is not close to complete. The Census of Marine Life estimated that somewhere between 500,000 and 700,000 marine species remain to be discovered and named. Subsequent revisions of that estimate, incorporating new data from environmental DNA surveys and molecular phylogenetics, have not reduced it. Some researchers believe the true number is higher. The pipeline from discovery to formal description is constrained by the number of trained taxonomists with expertise in deep-sea groups, which is a relatively small community, and by the funding available for the descriptive taxonomic work itself, which is rarely the most competitive use of research funds in a scientific funding environment that favors molecular biology, ecological modeling, and conservation application over specimen-based species description. The backlog of collected but undescribed specimens in museum collections worldwide is estimated at tens of millions of individual specimens. The organisms in those collections were collected, in many cases, decades ago. They are waiting for someone with the appropriate expertise and the time to look at them, write a description, choose a name, and submit it for publication. That process for a single species typically takes months to years. The backlog, at current rates of descriptive work, would take centuries to clear if it did not continue to grow. So when we ask what is really living in the deep ocean that science has not yet named, the honest answer is we do not know. We know that it is a lot. We know that it spans every major taxonomic group from bacteria to vertebrates. We know that it includes organisms that have been seen once on ROV footage and never again, organisms that exist only as genetic signals in environmental water samples, organisms sitting in museum jars labeled with collection date and location but no species name, and organisms in zones of the ocean that no sampling equipment of any kind has ever entered. The deep ocean covers more than half the surface of the planet. Its average depth is 3,700 meters. The vast majority of its volume has never been directly observed. The technology to observe it exists in the form of ROVs, landers, and deep-sea submarines, but deploying that technology requires ships, ship time, trained personnel, and funding, all of which are limited. What we have learned about the deep ocean in the past century has been extraordinary. The hydrothermal vent ecosystems, the deal vertical migration, and the biological carbon pump, the chemosynthetic communities of cold seeps and whale falls, the astonishing longevity of certain deep-sea organisms, the isolation and independent evolution of hadal faunas in separate trench systems. Each of these discoveries has changed not just marine biology, but our broader understanding of what life is capable of. And each discovery has been accompanied by new questions that the discovery itself generated. The answer to what is down there has not converged on a complete picture. It has expanded. Each expedition that returns with data about the deep ocean returns also with new observations that don't fit the existing picture, new species, new behaviors, new sounds, new ecological relationships that weren't predicted from what was known before. The deep ocean keeps its own inventory. It has been compiling that inventory for billions of years, without our input, without our categories, without our need for names. It is patient in a way that human science, with its funding cycles and career timelines and equipment replacement schedules, is not. The organisms in the deep ocean do not need to be named to be real. They do not need to be studied to be ecologically functional. They are simply there, in the cold and the dark and the pressure, doing what the conditions of their existence have shaped them to do, whether or not anyone is watching. And tonight, in the quiet of where you are, it is worth holding that thought. Not as something unsettling, but as something true. The planet is larger and stranger and more alive than any map of it suggests. Somewhere below the surface of the ocean, right now, something that no one has ever seen is going about its business in the way it always has. The deep scattering layer is rising toward the surface in the dark hours. The SOFAR channel is carrying sounds from one end of the ocean to the other. The Greenland shark is moving through cold Arctic water that may have been at the surface before any living human being was born. The snailfish of the Mariana Trench is somewhere near 8,000 meters, doing what snailfish at 8,000 meters do. The ocean is full of things we have not named. It has always been full of things we have not named. And the naming, when it comes, will be the beginning of the understanding, not the end of the mystery. The brine pools of the deep Gulf of Mexico offer one of the most visually dramatic and biologically puzzling features of the deep ocean. These are bodies of water sitting on the seafloor that are so saturated with dissolved salt that they are denser than the surrounding seawater. They do not mix with the water above them. They have a distinct surface, a sharply defined boundary at their edge, a shoreline of sorts. Creatures that enter them are typically killed by the osmotic shock of the hypersaline environment. And yet, at the edges of these pools, chemosynthetic communities thrive. The brine itself, in the largest pools like Orca Basin in the Gulf of Mexico, which reaches depths of around 2,400 meters, extends across hundreds of square meters of seafloor. The boundary between the brine pool and the surrounding seawater creates a halocline so sharp that ROV cameras have filmed the reflective surface of a brine pool looking almost exactly like a lake surface viewed from above. It is disorienting. There is a body of still, dense, lethally salty water sitting on the floor of the ocean, with its own distinct chemistry, its own bacterial community adapted to extreme salinity, and its own sharp boundary with the world around it. The organisms that live at the margins of brine pools, not inside them but at their edges, include tube worms, mussels with sulfide oxidizing bacterial symbionts, and a diverse community of fauna associated with the seeping hydrocarbons and hydrogen sulfide that maintain the chemical gradient at the pool's edge. Some of these organisms, particularly the tube worm species Lamellibrachia luimesi, are exceptionally long-lived. Individual specimens of Lamellibrachia luimesi, growing at cold seep sites in the Gulf of Mexico, have been estimated to live over 250 years, growing extremely slowly but persistently in an environment where food energy is available continuously from the chemosynthetic base. The slow growth and long life of many deep-sea organisms is a recurring theme in their biology. It is connected to the cold temperatures, which slow metabolic rates, and to the episodic or low-energy food supply, which constrains the resources available for rapid growth. The consequence is that deep-sea communities, particularly those associated with stable features like cold seeps and hydrothermal vents, can be very slow to recover from physical disturbance. This has implications for deep-sea mining, which is being explored commercially in the context of polymetallic nodules on the abyssal plain and the mineral deposits associated with hydrothermal vent systems. Polymetallic nodules, which are roughly potato-sized concretions of manganese, iron, nickel, copper, and cobalt that form very slowly on the abyssal floor over millions of years, are present in enormous quantities in certain parts of the Pacific. The Clarion-Clipperton Zone, an area of the Pacific between Hawaii and Mexico, contains nodule deposits that have attracted commercial interest. The organisms living on and around those nodules, and in the sediment between them, are part of a community that has developed over the same geological timescales as the nodules themselves. They are adapted to conditions of extreme stability, unchanging temperature, pressure, and chemistry over timescales far beyond any human reference. If the seafloor is disturbed by mining machinery, the sediment plumes generated can travel hundreds of kilometers and settle in areas beyond the direct mining zone. The organisms buried or smothered by those plumes do not recover quickly. Studies of natural disturbances to abyssal sediment, such as the trails left by whale carcasses that dragged along the seafloor, show that these tracks can remain visible in the sediment for decades. In 1989, as part of a study called the Deep Sea Impact Experiment Study, or DISCOL, German researchers deliberately disturbed a 10.8 square kilometer area of the Peru Basin abyssal plain using a trawl sled dragged repeatedly across the seafloor. They then monitored the site over subsequent years and decades. A revisit 26 years later found that the disturbed tracks were still visible in the sediment, that the macrofaunal community in the disturbed area had not returned to its pre-disturbance composition, and that the full biological recovery of the site was still incomplete a quarter century after the original disturbance. This is the pace of biological time in the deep ocean, not years, not decades, potentially centuries for full recovery. And it is the context in which all the unnamed organisms of the deep sea exist, an environment of extreme stability and extreme slowness, where evolution has had time to produce extraordinary specialization, but where ecological recovery from disturbance takes longer than any management cycle that human industrial planning currently incorporates. The whale fall community deserves more detail than we gave it earlier, because it is a remarkable example of how the deep ocean uses resources that surface biology generates. When a large whale dies in the open ocean, its carcass sinks. The descent can take weeks, during which the carcass passes through multiple depth zones, each with its own community of scavengers. The specific trajectory of the sinking carcass, the depth at which it eventually comes to rest, and the chemical composition of the seabed at that location all influence what happens next. In the first phase of decomposition, called the mobile scavenger stage, large mobile predators and scavengers dominate. Sleeper sharks, hagfish, rat-tail fish, and large crustaceans arrive rapidly, drawn by the chemical plume of the decomposing whale. They consume the soft tissue in a period that can last from months to years, depending on the size of the whale and the density of the local scavenger community. A large blue whale, which can weigh 150 tons, provides an enormous resource pulse to the surrounding environment. In the second phase, the enrichment opportunist stage, smaller organisms colonize the exposed bones and the surrounding sediment enriched by the decomposition products. This community is dominated by polychaete worms, crustaceans, and various mobile invertebrates attracted by the altered chemistry of the sediment near the carcass. In the third phase, the sulfophilic stage, the bones themselves become the primary resource. The fats preserved within the bone matrix begin to break down, producing hydrogen sulfide as a byproduct. This sulfide supports a chemosynthetic community structurally similar to that of a cold seep, bacterial mats, Osidax worms, mussels and clams with sulfide-oxidizing symbionts, and the broader community that these primary producers support. A single large whale carcass can support a chemosynthetic community for 50 to 100 years. This is the basis for Craig Smith's whale fall hypothesis, published in 1989, which proposed that whale fall communities could serve as stepping stones connecting hydrothermal vent and cold seep communities across the abyssal plain, allowing the specialized organisms of vent communities to disperse across the ocean floor between isolated vent fields. The genetic structure of vent species populations across multiple vent fields has been used to test this hypothesis, with mixed results. Some vent species do appear to use whale fall communities as intermediate habitats during dispersal. Others do not. The whale fall stepping stone hypothesis appears to be valid for some taxonomic groups and not others, and the relative importance of whale falls, cold seeps, and other organic falls as dispersal corridors varies by species and by ocean region. What this means for the taxonomy and biogeography of chemosynthetic communities is that the deep sea biological map is more connected, and the connections are more complex than the simple picture of isolated vent fields would suggest. And the detailed mapping of those connections, who lives where, who is related to whom, which populations interbreed, and which are reproductively isolated, is an ongoing program of research that will take decades to complete even with current technology. The pressure-adapted proteins of hadal organisms are a window into one of the more fundamental questions that deep-sea biology raises. What are the limits of life? The concept of the extremophile, an organism adapted to conditions that would be lethal to most life forms, has expanded dramatically since the discovery of hydrothermal vent communities. The range of conditions under which life has been found to exist has expanded with each decade of deep-sea research. Piezophiles, the pressure-loving organisms of the deep ocean, can function at pressures that would denature the proteins of surface organisms within minutes. The specific adaptations that allow this include changes to the amino acid sequences of key enzymes, changes to membrane lipid composition that maintain appropriate fluidity under pressure, and the accumulation of piezolites like TMAO that stabilize protein structure under elevated pressure. Some of these adaptations have been characterized in detail in the model Piezophile Photobacterium profundum, a bacterium isolated from Suruga Bay at 2,500 meters depth that can grow across a wide range of pressures, including the highest pressures that laboratory equipment can generate, which are equivalent to depths greater than any trench currently measured on Earth. The genome of Photobacterium profundum has been sequenced and compared with closely related surface bacteria, and the differences identify specific genes involved in pressure adaptation, providing a genetic basis for understanding how organisms become piezophilic. But Photobacterium profundum is a cultivated laboratory organism. Most piezophilic bacteria from the deep ocean cannot be cultivated in the laboratory at all, because the conditions required for their growth, not just pressure but the precise chemical composition of deep-sea water and sediment, cannot be replicated in a laboratory setting with current technology. These organisms are detected by environmental sequencing. Their genes are known, their presence in the deep ocean is established, but their physiology and ecology cannot be directly studied because they will not grow outside their natural environment. This is a general problem in deep-sea microbiology. The vast majority of deep-sea bacteria and archaea identified through environmental sequencing surveys cannot be cultured in the laboratory. They exist as genetic sequences in databases, with inferred metabolic capabilities based on comparative genomics, but without the direct experimental access that laboratory cultivation would provide. The uncultured majority of deep-sea microbes, which is to say most of the deep-sea microbial community by species count, remains largely uncharacterized at the physiological level. The archaea are worth singling out here. The archaea are a domain of life, distinct from both bacteria and eukaryotes, that was not recognized as a separate domain until Carl Woese and George Fox published their landmark work on ribosomal RNA in 1977. Before that work, archaea were classified as bacteria because they are single-celled organisms without nuclei and appear superficially similar to bacteria. The ribosomal RNA analysis showed that they are as different from bacteria at the molecular level as bacteria are from humans. Archaea include many extremophile groups: the thermophiles of hydrothermal vents, the halophiles of hypersaline environments, the methanogens of anaerobic sediments. In the deep ocean, archaea are particularly abundant in sediment, where they make up a substantial fraction of the total microbial biomass. Their specific roles in deep-sea biogeochemistry, the cycling of nitrogen, sulfur, carbon, and other elements in deep-sea sediment, are areas of intensive current research. One group of archaea, the Thaumarchaeota, are among the most abundant organisms in the mesopelagic and deep ocean by cell count. They are autotrophic, meaning they fix carbon from carbon dioxide using energy from ammonia oxidation, a process called ammonia oxidation or nitrification, which contributes to the transformation of nitrogen compounds in the ocean. The total amount of carbon fixed by Thaumarchaeota in the dark ocean below the photic zone has been estimated to be globally significant, on the order of the contribution from surface phytoplankton in some estimates. But those estimates are based on indirect measurements and carry substantial uncertainty. The sperm whale deserves particular attention in any discussion of what is unknown in the deep ocean, because the sperm whale is our indirect sensor, a biological probe that dives into zones we cannot easily reach and returns with evidence of what it found there. Sperm whales routinely dive to depths of 1,000 meters in search of prey, primarily squid. Their dives can last over an hour. The deepest confirmed sperm whale dives, based on acoustic tracking data and stomach contents, suggest they occasionally reach depths of 2,000 meters or more. They navigate and hunt in the complete darkness of the bathypelagic zone using echolocation. They produce clicks of extraordinary intensity, the loudest sounds produced by any animal, and interpret the returning echoes to locate and track prey. What they find at those depths, the prey they actually consume, is not directly observable by humans. We know what sperm whales eat primarily from the stomach contents of dead or stranded animals, from the beaks of cephalopods that accumulate in the stomachs and intestines and are slow to digest, and from the fecal material of living whales, which can contain squid beaks, fish otoliths, and other hard parts of prey. The squid beaks found in sperm whale stomachs include many that cannot be matched to any described squid species. These are beaks of unknown origin, from animals that have never been described, caught only in the digestive system of a whale that dove to depths we cannot easily follow. In the case of the colossal squid, the first indication that such a large animal existed in the Southern Ocean came from a sperm whale stomach containing a beak far larger than any previously cataloged squid beak. The animal that produced the beak was eventually found and named, but only years later. The sperm whale knew about it first. This pattern of discovery through stomach contents is not unique to squid. The beaked whales, which are among the deepest diving marine mammals with confirmed dives exceeding 2,000 meters and possibly reaching 3,000 meters, are even less well studied than sperm whales in terms of their diet and the organisms they encounter during their dives. New beaked whale species have been described as recently as 2016, when Berardius minimus was identified from specimens in Japan. Some beaked whale species are known primarily from stranded carcasses, with no confirmed living observations. Their behavior at depth, what they encounter and eat during dives that can last over 3 hours, is almost entirely unknown. The diversity of the beaked whales itself illustrates the general principle. There are approximately 23 described species of beaked whale. Some are represented in scientific collections by only a handful of specimens. The geographic distribution of some species is known only from the locations of stranded carcasses, which are a biased sample of where the animals actually occur, because strandings happen near coastlines and the animals range across open ocean. The ecology of most beaked whale species is poorly characterized, and their role in the deep-sea food web, including what specific prey they consume at depth, is incompletely understood for virtually all species. The cephalopods as a group represent one of the largest blind spots in deep-sea biology. Squid, octopus, nautilus, and cuttlefish collectively contain roughly 800 described living species. Estimates of undescribed species vary widely, but consistently suggest the true number is significantly higher. The deep ocean is particularly poorly sampled for cephalopods because conventional trawl nets badly underestimate their abundance, and because their soft bodies, which lack the hard parts that preserve well in trawl samples, leave little trace when the nets are hauled to the surface. The giant squid, Architeuthis dux, is the most famous example of a deep-sea cephalopod that is well documented in terms of its existence, poorly documented in terms of its living biology. The giant squid can reach lengths of 12 to 14 meters, including its tentacles. It is found in all the world's oceans at depths typically between 300 and 1,000 meters. It has been the subject of human fascination for centuries, inspiring the Scandinavian legend of the Kraken, appearing in historical accounts from multiple seafaring cultures, and generated more scientific literature than almost any other deep-sea invertebrate. And yet, the first confirmed footage of a living giant squid in its natural habitat was not captured until 2004. A Japanese research team, led by Tsunemi Kubodera of the National Science Museum in Tokyo, used baited hooks on a long line deployed at 900 meters depth near the Ogasawara Islands and recovered a series of still photographs from an automatic camera that showed a giant squid attacking the bait, becoming snagged on the hook and ultimately losing a tentacle before escaping. The tentacle, which was over 5 meters long, confirmed beyond any doubt that the photographs showed a living giant squid. But those photographs were stills. not video, and they showed the squid for only a brief period. In 2006, the same team, using a small ROV and a bioluminescent jellyfish as a lure, captured the first video footage of a living giant squid in the wild. The footage showed an animal approximately 8 meters long moving through the dark water with evident power. In 2019, another team, using a specialized camera system designed to operate with dim red light to avoid startling deep-sea organisms, filmed a giant squid in the Gulf of Mexico at approximately 760 meters depth. These observations confirm that the giant squid is real, that it lives in the mesopelagic and upper bathypelagic zones, and that it is capable of rapid movement when provoked or hunting. They do not tell us how giant squid reproduce, how long they live, how they communicate, whether they are solitary or gather in groups, what their acoustic environment is like from their perspective, or how many of them exist globally. The population size of Architeuthis ducks has never been estimated with confidence. The species is cosmopolitan, appearing to range across all ocean basins, but whether it is one globally distributed species or several reproductively isolated populations with distinct characteristics remains a subject of genetic debate. The problem of population structure in deep-sea species is broader than just the giant squid. For many deep-sea fish and invertebrates, the geographic range of a species has been established from the locations where specimens have been collected, but whether those specimens represent a single interbreeding population or multiple distinct populations is often not known. The deep ocean appears to offer pathways for dispersal, particularly through the deep scattering layer and the midwater currents that move organisms horizontally over large distances. But physical barriers can also exist in the deep ocean, particularly in the form of the mid-ocean ridges that divide ocean basins. The Loihi Seamount, off the coast of Hawaii, is an active submarine volcano currently building toward the ocean surface. It sits at a depth of about 1,000 meters at its summit. The hydrothermal systems associated with Loihi have been studied since the 1980s and have revealed a unique microbial community adapted to the unusual iron-rich chemistry of the fluids venting from this volcano. The Loihi microbial communities include iron-oxidizing bacteria that are among the most efficient iron oxidizers known, producing an orange, fluffy material called iron snow that drifts down from the vent fields in billowing columns. The iron-oxidizing bacteria of Loihi are not typical of most hydrothermal vent systems, which are dominated by sulfur-based chemistry. Loihi represents a different chemical regime, producing a different community, and serving as a model for understanding how microbial communities might function in environments with high iron and low sulfur, which are relevant to certain hypothetical subsurface ocean environments on other planets. Every submarine volcano, every seamount, every geographic feature of the deep seafloor that creates local variations in chemistry, pressure, or temperature is a potential site of biological distinctiveness. There are estimated to be over 100,000 seamounts in the global ocean, defined as underwater mountains rising at least 1,000 meters from the surrounding seafloor. Of those, fewer than 1,000 have been biologically surveyed in any detail. Each systematically surveyed seamount has produced endemic species, organisms found nowhere else, specifically adapted to the particular conditions of that seamount. The pattern is consistent enough that seamount endemism is now treated as a general principle rather than a surprising finding. But the implication of that principle, when applied to the tens of thousands of unsurveyed seamounts, is that the amount of biological diversity sitting undiscovered on the summits and slopes of unsurveyed seamounts alone is very large. The deep-sea coral communities associated with many seamounts amplify this further. Cold water corals, which do not require sunlight and build reefs from calcium carbonate in deep cold water from depths of 40 to 6,000 meters, are among the most structurally complex habitats in the deep ocean. They provide shelter and attachment surfaces for hundreds of associated species of sponges, worms, crustaceans, and fish. The deep-sea coral reefs off Norway, Ireland, and New Zealand are among the best studied, and in all of these systems, regular biological surveys continue to add new species to the known fauna of the reef. The oldest individual cold-water coral colonies that have been dated using radiometric methods are over 4,000 years old. They are among the oldest living structures built by animals on the planet. These ancient coral colonies grow at rates of millimeters to a few centimeters per year. They are vulnerable to physical disturbance from bottom trawling, which has destroyed large areas of cold-water coral habitat in European and Pacific waters over the past 50 years. The biological recovery of a destroyed cold-water coral reef, given the growth rates of the corals themselves, would take thousands of years. Reefs that have been trawled repeatedly in recent decades will not return to their pre-disturbance state within any human timescale. The question of how deep-sea organisms communicate, and whether communication is the right word for what they do, is a productive place to spend some time. For surface organisms, communication involves visual signals, acoustic signals, chemical signals, and in some species, electrical signals. All of these channels are available in the deep ocean, but their relative importance is shifted dramatically by the absence of light and the very different acoustic environment. Chemical communication, mediated by dissolved compounds detected by chemoreceptors, appears to be particularly important in the deep ocean. It is chemosensory detection that brings the hagfish, the sleeper shark, and the amphipod to a whale fall within hours of the carcass arriving on the seafloor. The chemical plume from a decomposing whale can travel with deep ocean currents for kilometers, and the organisms that detect it must be able to extract directional information from a gradient in dissolved concentration to navigate toward the source. Hagfish are remarkable examples of a deep-sea organism whose sensory biology is adapted to this chemical world. They are jawless fish belonging to the oldest surviving lineage of vertebrates, essentially unchanged in fundamental anatomy for 300 million years. They have very poor eyesight, a vestige of a more functional visual system in their ancient ancestors. What they have instead is an extraordinary chemosensory system, distributed across their bodies in a dense array of receptor cells. They can detect and navigate to decomposing organic material at very low concentrations. Hagfish are also notable for their defense mechanism. When threatened, they produce enormous quantities of a fibrous, gel-like slime that expands in water to fill a remarkable volume. A single hagfish can produce enough slime to fill a 20-liter bucket in seconds. The slime is composed of mucin proteins and silk-like threads, and it works by clogging the gills of predators that try to swallow the hagfish. The predator, unable to breathe through slime-filled gills, releases the hagfish. The hagfish then removes the slime from its own body by tying itself into a knot and sliding through the knot, a behavior called knotting. The biomechanics of hagfish slime have attracted interest from materials scientists because the silk-like threads within the slime are among the strongest natural fibrous materials known, comparable in tensile strength to spider silk. Research into the molecular structure and production mechanism of these threads is ongoing, with potential applications in materials engineering. The hagfish, a 300-million-year-old jawless scavenger of the deep sea floor, is thus simultaneously a subject of marine biology, evolutionary paleontology, and materials science. The sea spiders, or pycnogonids, are another group that illustrates the breadth of what the deep ocean contains. Pycnogonids are marine arthropods, distantly related to but distinct from terrestrial spiders, with a body plan that is essentially a small central body from which multiple long legs project. In shallow water, pycnogonids are typically small, a centimeter or less. In the deep ocean, they follow the pattern of gigantism. Antarctic and deep-sea pycnogonid species can reach leg spans of over 70 centimeters. They move slowly across the seafloor and on the surfaces of corals, feeding on soft-bodied invertebrates. Their internal anatomy is unusual even by arthropod standards. The gut extends into the legs, and in some species, even the gonads extend into the leg segments. There are approximately 1,300 described species of pycnogonid globally. Their diversity in the deep ocean is poorly known, partly because their fragile legs are damaged or lost during trawl collection, making specimen identification difficult from damaged material. ROV footage of Antarctic and deep-sea pycnogonids has shown a diversity of forms and sizes that does not always correspond to the described species catalog, suggesting that a significant fraction of deep-sea pycnogonid diversity remains undescribed. The polychaete worms of the deep seafloor are probably the most species-rich large-bodied group in abyssal sediments and among the least known. Polychaetes are segmented marine worms belonging to the class Polychaeta, which contains some 10,000 described species globally. They occupy every marine habitat, from intertidal zones to the deepest trenches, and play key roles in sediment bioturbation, organic matter processing, and as prey for other organisms. In the abyssal sediment, polychaetes are consistently the most abundant macrofaunal organisms recovered from sediment cores in terms of both individual animals and species diversity. The number of undescribed polychaete species from the deep ocean is thought to be very large. Every systematic sampling program of abyssal sediment that has been analyzed in detail has found polychaete morphospecies, meaning physical forms distinguishable from each other but not yet formally described, that significantly outnumber the described species from the same habitat. In one analysis of abyssal sediment samples from the Pacific, researchers estimated that the total polychaete diversity of the abyssal Pacific alone might be in the thousands of species, the majority of which are undescribed. This is a consistent finding across deep-sea sediment surveys. The diversity of small organisms in the sediment, the meiofauna and macrofauna living in and on the sediment surface, is consistently higher than the described species catalog would suggest, and the discrepancy increases with depth and distance from areas that have been studied previously. The deep ocean is not a place where species diversity decreases as you go deeper in a simple linear way. The relationship between depth and diversity is complex, varies by taxonomic group, and is not fully characterized. The nematodes are worth mentioning in this context because they represent the most abundant multicellular animals on the planet by individual count, and a substantial fraction of the individuals on Earth live in deep-sea sediment. Nematodes are microscopic roundworms, typically less than a millimeter long, that are found in virtually every environment on Earth: agricultural soil, freshwater sediments, plant roots, the bodies of other animals, and the sediment of the deep ocean. In the deep sea, nematode densities can reach millions of individuals per square meter of sediment. The species diversity of deep-sea nematodes is essentially unknown. Estimates of the total number of nematode species globally vary by an order of magnitude depending on the method of estimation, from tens of thousands to over 1 million. For deep-sea nematodes specifically, systematic surveys are sparse enough that a reliable diversity estimate is not available. The fungi of the deep ocean have only recently begun to receive systematic attention. Fungi are not typically thought of as marine organisms, and for most of the history of marine biology, they were considered minor components of the marine ecosystem. Molecular surveys of deep-sea water and sediment have revealed that fungi are present throughout the deep ocean, including at abyssal and hadal depths, in a diversity that was not anticipated. The ecological roles of deep-sea fungi are not well characterized. They may participate in the decomposition of organic material, in the biological modification of minerals, or in symbiotic associations with other deep-sea organisms. This is an early-stage research area in which the basic inventory of what species are present is still being established. All of this, the nematodes, the pycnogonids, the polychaetes, the fungi, the unculturable bacteria and archaea, the undescribed cephalopods and siphonophores, the organisms detected by environmental DNA but never seen, the organisms filmed once and never identified, constitutes a picture of the deep ocean as a place where our knowledge is genuinely partial, not uncertain in the way that quantum mechanics is uncertain. Genuinely partial in the simpler sense that we have not looked or have not been able to look carefully enough, and the things we have not seen are simply out there, continuing to exist without requiring our knowledge of them. The pace of discovery in deep-sea biology has not slowed since the census of marine life. If anything, advances in technology have accelerated it. Environmental DNA sequencing, which can be done on seawater samples collected by autonomous vehicles without any specialized biological expertise, has expanded the geographic scope of surveys dramatically. Autonomous underwater vehicles that can operate for weeks without ship support have extended the reach of acoustic and optical surveys into areas previously accessible only by manned submersibles or tethered ROVs. Machine learning tools applied to the massive archives of underwater imagery collected over decades are identifying organisms that human reviewers missed or could not classify. These tools are generating new data faster than specialists can analyze it. The pipeline between data collection and biological understanding is backed up, not because researchers are slow, but because the volume of new data is large and the number of specialists who can interpret it for specific taxonomic groups is small. There is a working estimate used in some marine biodiversity discussions that suggests we have described something like 30 to 40% of the total species diversity of the ocean. That estimate is rough and contested. Some researchers think it is optimistic. What it suggests, if it is even approximately correct, is that the majority of the species that live in the ocean have not yet been described. They have not been named. They have not been studied. They exist in the ocean, playing their ecological roles, interacting with each other and with the organisms we have named, persisting through conditions that would be lethal to us. and the fact of their existence is currently recorded nowhere except in the biology of the ocean itself. This is not a failure, it is a description of the situation. The ocean is large, it is old. It has been generating biodiversity for over 600 million years. The tools to study it systematically at depth have existed for only a few decades. The resources devoted to that study are substantial in absolute terms, but modest relative to the scale of the task. When the first hydrothermal vent communities were found in 1977, they changed the definition of what life requires to exist. When the mesopelagic biomass estimates were revised in 2014, they changed the calculations for the global biological carbon pump. When Osidax worms were discovered in 2002, they revealed an ecological role that no one had predicted. Each of these discoveries was made in the context of a research community already convinced that deep-sea exploration was scientifically productive, and each of them generated new questions that will take years or decades to answer. The deep ocean is not a solved problem. It is not a frontier that has been crossed and left behind. It is an ongoing scientific encounter with a part of the planet that is enormous, complex, ancient, and largely beyond direct human experience. The things in it, the named things and the unnamed things and the things that have been glimpsed once and not seen again, continue to exist on their own terms, in conditions that were old when human civilization was young. There is something in that which invites a particular kind of quiet attention, not awe in the theatrical sense, a steadier thing. The recognition that the world is considerably larger than any map of it, and that most of what is in it is getting along perfectly well without our knowledge of it. Consider for a moment the coelacanth, because the coelacanth is the clearest demonstration in living biology of how wrong the assumption of completeness can be. The coelacanth is a large, deep-bodied fish with lobed fins, belonging to a lineage that was thought to have gone extinct 65 million years ago. In the same mass extinction event that ended the non-avian dinosaurs, the coelacanths were known from fossils, widespread in the record, present across millions of years of geological time, and then apparently absent from any deposits younger than the end Cretaceous boundary. The prevailing scientific consensus, held with confidence for more than a century, was that they were extinct. In December 1938, a South African museum curator named Marjorie Courtney Latimer was examining the catch from a local fishing trawler in East London, South Africa. Among the fish, she found one she did not recognize. Large, bluish, roughly 1.5 meters long, with unusual fleshy fins unlike any fish she had seen. She preserved it and sent a description and sketch to JLB Smith, a South African ichthyologist. Smith recognized it immediately from the fossil record. It was a coelacanth. It had been hauled up from approximately 70 meters depth in the Chalumna River estuary. The trawler had been operating in an area that was not considered deep sea by any conventional classification. The fish was real. It was alive. Its lineage had survived, not in the fossil record, but in the actual ocean, for 65 million years after it was supposed to have been eliminated. The scientific name of this species, Latimeria chalumnae, honors both the curator who preserved it and the river estuary where it was found. The species was and remains one of the most celebrated discoveries in the history of zoology. A second living coelacanth species, Latimeria minatoensis, was discovered in Indonesia in 1998, identical in general appearance to the African species, but genetically and geographically distinct. Both living coelacanth species are found at depths of 150 to 400 meters, in submarine caves and along steep rocky slopes adjacent to deep water. They are nocturnal, spending their days in the caves and emerging at night to drift with the current and feed. Their feeding behavior is passive. They orient themselves in various postures, including occasionally vertical and upside down, and wait for small fish and cephalopods to come within striking range. They do not swim actively in the way that most bony fish do. Their locomotion uses a diagonal pattern of fin movement more similar to a tetrapod walking than to conventional fish swimming, which has attracted intense interest because the lobed fin relatives of the coelacanths are thought to be close to the lineage from which terrestrial vertebrates ultimately evolved. The discovery of the coelacanth did not happen in a remote, inaccessible part of the ocean. It happened in a trawl net off the coast of South Africa, in water less than 100 meters deep, in a location that had been fished for decades. The fish existed. Fishermen in the Comoros Islands, where coelacanths are relatively more common, knew of them and occasionally caught them. calling them gumbessa. The information simply had not reached the formal scientific literature in a way that prompted investigation. This points to a dimension of the unknown in marine biology that is separate from the purely technological limitation. Some of what is unknown is not out of reach because we lack the technology to reach it, but because the information exists in local knowledge, in fishing communities, in oral traditions, in records not integrated into the formal scientific literature, and has not been recognized as scientifically significant by the people who encountered it. Many deep-sea organisms that are now formally described were known for years or decades to fishermen who caught them in trawl nets, saw them at depth on bait lines, or found them stranded on beaches before they came to the attention of researchers who could give them formal names. The megamouth shark, a filter-feeding shark that can reach 5 meters in length and was a completely unknown genus and species, was discovered in 1976 when a U.S. Navy research vessel accidentally captured one in its sea anchor off Hawaii. The sea anchor was deployed at 165 meters depth. The megamouth was not a deep trench specialist. It occupied depth ranges that humans had been accessing for centuries. It was simply not recognized and described until the 1970s. Since its discovery, the megamouth has been encountered approximately 100 times in documented records, through strandings, incidental capture in fishing gear, and direct observation. Its diet is planktivorous, filtering small crustaceans and jellyfish from the water. Its behavior, including whether it migrates vertically like the organisms of the deep scattering layer, is not well characterized. Whether its global population is stable, declining, or in any particular trend is unknown, because there is no baseline estimate of its population size to compare against. The whale shark, the largest fish in the ocean, was not formally described until 1828, and was poorly understood for over a century after that. The basking shark, the second largest, was known to coastal communities in the British Isles for centuries, but was not scientifically described until 1765. Both of these are animals several meters long, found in surface waters accessible to humans for all of human history. Their existence was known. Their biology was not understood until formal science caught up with what fishing communities and coastal observers had been seeing for generations. The implication is not that science is slow or inefficient. It is that the ocean is large enough and its inhabitants numerous enough that even organisms large enough to be clearly visible and in habitats accessible to ordinary human activity can escape scientific description for extended periods. For organisms in the deep ocean, genuinely inaccessible to direct human observation without specialized and expensive equipment, the lag between existence and description is even longer. The genome of the nautilus, a group of cephalopods whose relatives have existed since the Cambrian period over 500 million years ago, was sequenced for the first time in 2015. The nautilus is not a rare or obscure organism. It is well known, frequently kept in aquariums, commonly described in popular science writing. Its shell has been collected and traded for centuries. And yet the complete genome sequence, which reveals the genetic architecture of how a nautilus builds itself, regulates its physiology, and relates to other cephalopods, was not available to researchers until a decade ago. For deep-sea species, the genomic information is far more sparse. The genome of the giant squid was published in 2020 by a team that worked with tissue samples from multiple individuals, including a fresh specimen found stranded in Newfoundland. The giant squid genome is large, over 2.7 billion base pairs, and contains a number of interesting features, including an unusually large number of protocadaran genes, which in other animals are associated with the development of complex nervous systems. The giant squid has a large and anatomically complex brain. What it does with that brain, how it processes information, how it navigates, how it interacts with other individuals of its kind, remains essentially unknown because the animal has never been studied alive in its natural environment for more than the brief moments captured on camera. Octopus behavior, by contrast, is relatively well studied because octopuses can be maintained in aquariums and observed in controlled conditions. They demonstrate problem-solving, tool use, individual personality differences, and a remarkable capacity for camouflage that involves real-time neural control of chromatophores, the pigment-containing cells in their skin. But nearly all of this research has been done on shallow water species. The deep-sea octopuses, which include the Dumbo octopuses of the genus Grimpoteuthis, named for the ear-like fins they flap to swim, are known primarily from occasional ROV footage and fragmentary specimens. Whether deep-sea octopuses exhibit the same cognitive flexibility as shallow water species is unknown. The question cannot currently be investigated because the animals cannot be maintained in laboratory conditions at the temperatures and pressures of their natural habitat. The midwater cephalopods in general, the squid that inhabit the mesopelagic and bathypelagic zones, represent a substantial fraction of the total cephalopod diversity and are among the least well-studied groups. Some families, like the glass squid family Cranchidae, are known from relatively many preserved specimens because they are sometimes caught in relatively large numbers in mid-water trawls. Others, like the ram's horn squid Spirula spirula, whose internal shell is the most frequently found cephalopod shell on beaches worldwide, despite the animal itself being rarely seen, are known in extraordinary detail from their hard parts and fragments, but almost not at all from direct observation of the living animal. The ram's horn squid lives in the mesopelagic zone at depths of 200 to 1,000 meters. It is the only living member of its order. It has a coiled internal shell which is gas-filled and functions as a buoyancy organ. When the squid dies, the shell floats to the surface and is distributed by currents, washing up on beaches across the tropics and subtropics in enormous numbers. Beachcombers in the Caribbean, along the coasts of southern Africa, and on Pacific islands commonly find these white spiral shells, which look like miniature nautilus shells, without knowing what animal they came from. The living animal has been seen only a handful of times in brief ROV encounters, and its detailed biology is poorly known. The meso- and bathypelagic fish families beyond the lanternfish and hatchetfish already discussed include groups like the barreleye fish. The barreleye, of the family Opistoproctidae, carries cylindrical tubular eyes pointing upward, looking for the silhouettes of organisms above them. What makes them famous in popular science coverage is the transparent head, a fluid-filled cavity of transparent tissue through which the tubular eyes are visible from the outside. This transparent structure was known from preserved specimens, in which the tissue typically becomes opaque. The first clear demonstration that living barreleye fish have transparent heads, and the first documentation of the full range of eye movement the fish uses, including rotating the eyes to point forward during feeding, came from Mbari ROV footage published in 2008. Before that footage, the function of the transparent head and the mobility of the eyes were not understood, because preserved specimens do not reveal these features. The fish was known. Its anatomy was partly described. The transparent head was noted as a structural feature in the description of preserved specimens. But what the living animal actually did with that anatomy, how it actually used its unusual eyes in the context of its behavioral ecology, was not known until the ROV footage provided the context. This is a third category of unknown. Separate from the unnamed species and the unnamed organisms detected by DNA, the known species whose biology is described from dead or captive specimens in ways that are fundamentally incomplete because the living animal in its natural context does something qualitatively different from what the preserved material suggests. For every barreleye, where the discrepancy was eventually resolved by ROV footage, there are likely many other species where the same discrepancy exists but has not yet been discovered because the living animal has not been observed in the right conditions at the right depth. The deep ocean, then, contains several overlapping categories of scientific unknown. There are species that are physically present and accessible but have not yet been formally described and named. There are species whose existence is detected through environmental DNA or acoustic signatures, but that have never been physically observed. There are species known from very few specimens whose biology is essentially uncharacterized. There are species known only from indirect evidence, through stomach contents of predators or fragments in sediment. There are behaviors and ecological interactions that have not been directly observed because the animals are in places where cameras have not been pointed. And there are descriptions of known species that are incomplete in ways not yet recognized because the discrepancy between the dead specimen and the living animal has not been discovered. None of these categories is small. Together, they constitute a very large fraction of the actual biological content of the ocean. When researchers deploy a new lander to a previously unsurveyed abyssal site, when an ROV cameras into a submarine canyon for the first time, when an autonomous glider collects water samples from the bathypelagic zone of a remote ocean basin, they are not exploring with any expectation of finding nothing. They consistently find things, new things, species not previously recorded at that location, behaviors not previously documented, ecological configurations not matching existing models. The deep ocean keeps being surprising, and the surprise is not diminishing with time and effort. It is, if anything, becoming more organized, more systematic, better characterized in its dimensions, but not smaller. The naming will continue. New species will be described. New genera will be erected. New ecological roles will be documented. The pipeline will process more slowly than the ocean generates material for it, but it will keep moving. And in the meantime, the mesopelagic zone is still rising in the dark, carrying its billions of unnamed passengers toward the surface for another night of feeding. The SOFAR channel is carrying sounds from one end of the ocean to another, some of them from animals we have described, and some from animals we have not. The xenophyophore on the abyssal plain is extending its cytoplasm through the sediment, doing what it has been doing for millions of years in the dark, in the cold, under pressure that would flatten a car, under no obligation to be known. The Greenland shark is somewhere in the cold, dark water of the North Atlantic, moving at its usual slow pace, carrying its decades or centuries in its body, its eye clouded by the copepod that has lived on it possibly for years. The colossal squid is in the Southern Ocean at depth, doing something that no camera has captured. The 52 Hz whale is producing its call somewhere in the Pacific, as it has been producing it for more than 30 years, into water that carries the sound but offers no reply. The ocean is patient. It was here before the first animals evolved in it. It will be here after the last human ship returns to port. It has been holding its inventory for far longer than anyone has been asking about it, and it will continue to hold what it holds with the same indifference that it has always had to human curiosity about it. That is not a diminishment. It is the actual scale of the thing. And lying where you are now, in the quiet, it is worth letting that scale be real for a moment. Not as a weight, as a fact. The ocean is large. It is full of things. Most of them do not have names yet. Most of them are doing fine. The term abyssal refers not just to a depth range, but to a quality of environment that has no real equivalent in human experience. The abyssal zone, from roughly 4,000 to 6,000 meters depth, occupies the deep basins of the ocean floors, the great flat expanses of sediment between the mid-ocean ridges and the continental slopes. The pressure there, between 400 and 600 atmospheres, the temperature near freezing, the food supply thin, and the light absolute zero. These conditions have been stable across geological time on a scale that surface environments rarely approach. This stability is itself biologically significant. It means that the organisms of the abyssal zone have not had to adapt to the kinds of environmental variability that drive much of the evolutionary change in surface organisms. Seasonal cycles, glacial cycles, the changing of ocean chemistry with changing atmospheric composition. These are things that the abyssal fauna experiences only in muted form, if at all. The result is that many abyssal organisms have very ancient origins, morphological features similar to fossil forms that lived tens of millions of years ago, evolutionary rates that are extremely slow by biological standards. The glass sponges, belonging to the class Hexactinellida, are among the most remarkable of these ancient deep-sea organisms. They build their bodies from a rigid skeleton of silica, the same material as glass, arranged in a lattice that can be extraordinarily geometrically precise. The Venus's flower basket sponge, Euplectella aspergillum, produces a skeleton of interlocking silica struts that has attracted considerable interest from engineers studying structural design principles. The skeletal architecture of Euplectella achieves a combination of lightness, strength, and flexibility that current human engineering struggles to replicate at comparable scales. Glass sponges live in water from about 200 meters to over 2,000 meters depth, with some species extending into abyssal depths. They filter feed, drawing water through their tissues to extract bacteria and dissolved organic material. They are among the longest-lived of deep-sea organisms. The structure of their silica skeleton continues to grow throughout life, and the largest specimens in certain cold, deep-water habitats have been estimated to be thousands of years old, based on their growth rates and body size. The glass sponge reefs of the northeastern Pacific, found primarily off the coasts of British Columbia and Alaska, are among the oldest living structures on Earth. They are built by several species of glass sponge whose dead skeletal material accumulates on the reef framework as living sponges continue to grow on top. The reefs in British Columbia have been dated to approximately 9,000 years before present, making them older than the Egyptian pyramids, older than Stonehenge, older than most of the civilizations whose ruins are considered ancient. These reefs were not discovered by science until 1987. Before that discovery, glass sponge reefs were known only from the fossil record, where they were documented as common features of Cretaceous oceans, 145 to 66 million years ago. The living reefs off British Columbia were therefore, like the coelacanth, a living relic of a biological community type that had been assumed extinct. They had been there for 9,000 years, in waters that are in some areas accessible to commercial fishing, without anyone having investigated them systematically enough to recognize what they were. The deep-sea echinoderms, the group that includes sea urchins, sea stars, brittle stars, sea cucumbers, and feather stars, demonstrate a similar pattern of ancient lineages persisting in the deep ocean, while their shallow water relatives have changed more substantially over geological time. The brittle stars in particular are among the most abundant and diverse echinoderms in the deep ocean, living at all depths from the intertidal to the hadal zone. Deep-sea brittle stars of the order Ophiurida include species with remarkable regenerative capabilities. They can voluntarily shed arms to escape predators and regrow the lost arms completely. Some species can survive the loss of all 5 arms simultaneously and fully regenerate. The regenerative biology of brittle stars has attracted interest in biomedical research for what it might reveal about the mechanisms of tissue regeneration in non-mammalian animals. But the deep-sea brittle star species most likely to yield useful information about extreme regeneration are, in most cases, poorly characterized at the level of their genome and molecular biology because they have not been targets of sustained research programs. The diversity of brittle stars in the deep ocean is genuinely extraordinary. A single dredge sample from the abyssal floor in a previously unsampled area regularly yields multiple brittle star species not previously recorded from that location. The global species count for deep-sea ophiuroids is not confidently established, and new species are described regularly from samples collected during deep-sea surveys. The flatfish of the deep ocean present another category of poorly known organisms. The Greenland halibut, Reinhardius hypoglossoides, is commercially valuable and relatively well studied by fisheries science standards at depths from 200 to over 2,000 meters in the North Atlantic and Arctic. But most deep-sea flatfish, particularly those in the family Pleuronectidae that inhabit truly abyssal depths, are known from very few specimens and have not been systematically characterized in terms of their behavior, diet, population structure, or ecology. The trilobite shrimp, belonging to the order Notostraca, survived the Permian mass extinction event 252 million years ago, which killed approximately 96% of all marine species. They were known from the fossil record as Paleozoic organisms. Living specimens were eventually found in temporary freshwater pools in various parts of the world, where they hatch quickly from drought-resistant eggs after rainfall fills the pools. They are not deep-sea organisms. They are mentioned here because they represent, like the coelacanth and the glass sponge reefs, a category of biological discovery that was once unimaginable and then became real. An organism extinct according to the fossil record, living today. Deep-sea biology has its own version of this category, and it is larger than the coelacanth alone. The Jurassic shrimp Anaspides tasmaniae lives today in fresh water in Tasmania, essentially unchanged from its fossil relatives dating to the Triassic period. The nautilus, already mentioned, is a living representative of a lineage known from fossils 500 million years ago. The horseshoe crab, Limulus polyphemus, is morphologically nearly identical to fossils from 450 million years ago. These are not deep-sea organisms, but the pattern they represent, the persistence of ancient lineages in stable environments, applies with even more force to the deep ocean, which is one of the most stable environments on the planet. The expectation that systematic deep ocean surveys will continue to find organisms belonging to ancient lineages, thought extinct from the fossil record, is not a fringe position. It is a reasonable inference from the history of deep-sea biology. The coelacanth was the most dramatic example. It may not be the last. The polar oceans deserve specific mention because they represent environments where the deep cold of the ocean reaches the surface, creating conditions at relatively shallow depths that resemble the deep sea in temperature and, in some ways, in biological character. The Antarctic continental shelf, which lies at depths of 400 to 800 meters due to the weight of the Antarctic ice sheet depressing the continent, hosts communities dominated by sponges, brittle stars, and isopods that would be unusual anywhere outside the deep ocean. The organisms in these communities, living in water at or near the freezing point, exhibit many of the biological characteristics of deep-sea organisms: slow growth, long lives, large body sizes in some groups, and poorly characterized ecology. When the Larsen B ice shelf in Antarctica collapsed in 2002, it exposed a seafloor that had been covered by ice for at least 12,000 years. Researchers who investigated the exposed seafloor in the years following the collapse found a community of chemosynthetic organisms associated with methane seeps from the organic material that had accumulated under the ice. The organisms in this community had established themselves in the brief period between the ice shelf collapse and the first scientific survey, within a few years. They demonstrated that deep-sea chemosynthetic communities can colonize suitable environments quickly, even in previously ice-covered polar waters. The scale of what remains unknown in the deep ocean is, in the end, a reflection of the scale of the ocean itself. The ocean covers 71% of the surface of the Earth. Its total volume is approximately 1.335 billion cubic kilometers. It has been a liquid water environment for at least 3.8 billion years, providing stable conditions for the evolution of life for longer than any other environment on Earth. The life it has generated over that time, the diversity it has produced and maintained, is proportionally large. Human systematic study of this environment has been underway in a rigorous scientific sense for less than 200 years, beginning with the voyage of HMS Challenger in 1872 and 1876, which was the first expedition specifically designed to study the physical and biological characteristics of the deep ocean. The Challenger expedition dredged samples from dozens of deep ocean locations, described hundreds of new species, and established that life existed at depths previously considered impossible. It is the foundation on which all subsequent deep-sea biology has been built. In the century and a half since the Challenger, the tools for deep-sea research have improved dramatically. But the fundamental situation has not changed. We are exploring a very large environment with limited equipment, limited ship time, and limited specialist expertise, and we are finding things faster than we can characterize and name them. That is the situation. It is also, in a specific way, a measure of what the ocean is, not a problem to be solved and set aside. An ongoing encounter with a part of the world that is genuinely large enough, genuinely complex enough, and genuinely old enough to continue producing surprises for as long as anyone chooses to look. Tonight, in the dark, in whatever quiet you have found, consider the deep scattering layer. Right now, across every ocean, billions of small fish are rising toward the surface. Lanternfish, hatchetfish, bristlemouths, species that are named and species that have no name. They are moving upward through hundreds of meters of dark water, following the same cue that their ancestors have followed for tens of millions of years. They do not know they are unknown to science. They do not know that there are researchers who have spent careers trying to understand their migrations. They are simply doing what the conditions of their existence have shaped them to do. The 52 Hertz whale is somewhere in the Pacific. It has been calling for over 30 years. No response has been recorded. Whether it is calling to communicate or whether the sound is simply what it produces, the ocean carries it regardless. The sound travels through the SOFAR channel. The instruments detect it. The researchers note it. The animal continues. Somewhere at around 8,000 meters in the Mariana Trench, a snailfish is doing what snailfish at 8,000 meters do. The pressure there is 800 atmospheres. The temperature is close to 2 degrees Celsius. The fish is pale. It moves slowly. It is one of the deepest vertebrates ever recorded. It is fully at home. It needs nothing from us. The xenophyophore on the abyssal plain is extending its cytoplasm through the sediment in the dark. It is a single cell. It is the size of a dinner plate. It has been there for longer than we have been looking. The glass sponge reef off British Columbia, 9,000 years old, is filtering water in the cold dark of the Pacific continental margin. The same thing it was doing when the last glaciers were retreating. The same thing it was doing before the first human settlement on that coast. The colossal squid is in the Southern Ocean at depth. It has never been observed alive. It is somewhere. It is large. It is doing something. The deep ocean keeps its own count. It does not require us to count along. But we keep counting. And every time we do, the number is larger than we expected. And the things at the edge of our knowledge are stranger than the things at the center. And the boundary keeps moving outward, not inward. The ocean is not getting smaller as we learn more about it. It is getting larger. It was always that large. We are just beginning to see how large. The bathyal zone, the region between approximately 200 and 4,000 meters depth, is where the majority of the described deep-sea macrofauna lives. It is deeper than the mesopelagic zone, but shallower than the abyssal, and in geographic terms it includes the continental slopes, the sides of seamounts, and the walls of mid-ocean ridges. This is the zone where the most deep-sea sampling has been done, because it is accessible to instruments deployed from standard research ships without the need for full ocean-depth vehicle systems. Even in this relatively accessible zone, the biodiversity is substantially underdescribed. The continental slopes of the world's oceans cover an area comparable to the combined surface area of all the world's continental landmasses. The species that live on the slopes of one ocean basin are often not the same as those on the slopes of another, and within a single ocean basin, species composition changes with depth, latitude, and the local oceanographic and geological conditions. The deep-sea fish families of the bathyal zone include the macrorids, or rat-tail fish, which are elongated, large-headed, tapering-tailed fish that are among the most abundant large fish on the continental slopes of all ocean basins. There are approximately 300 described species of macrorid. They are important parts of the deep-sea food web, feeding on invertebrates and smaller fish, and being fed upon in turn by sharks and other predators. Their diversity on unexplored slope habitats is not well characterized, and new species are described regularly from deep surveys. The chimeras, an ancient group of cartilaginous fish related to sharks and rays, are another bathyal family with many poorly characterized species. There are roughly 50 described species of chimera, distributed across all ocean basins. They typically live on or near the seafloor at depths from a few hundred to several thousand meters. Their biology, beyond what can be inferred from dead specimens, is in most cases not known. Their reproductive biology is unusual for cartilaginous fish. They lay eggs in tough, horny cases deposited on the seafloor, and the developing embryo takes months to years to complete development within the egg case. The specifics of this process, including how long eggs take to hatch at different depths and temperatures, have been documented for only a few species. The deep-sea sharks deserve attention because they are simultaneously among the most poorly understood large vertebrates in the ocean and among the most commercially significant in terms of bycatch in deep-sea fisheries. The family Squalidae, the dogfish sharks, includes numerous deep-water species. The family Etmopteridae, the lantern sharks, contains over 50 species, the majority of which are small, bioluminescent, and mesopelagic or bathypelagic. Lantern sharks use their bioluminescent photophores for counter-illumination, like the hatchetfish, and possibly for intraspecific communication. In some species, the photophore pattern on the flank appears to be species-specific and sex-specific, suggesting a signaling function. The behavioral evidence for this inference is indirect because lantern shark behavior has not been directly observed in the wild in any systematic way. The Pacific sleeper shark, Somniosus pacificus, a close relative of the Greenland shark, ranges from the Arctic into the Pacific tropics at depths from shallow waters to over 2,000 meters. Like the Greenland shark, it is thought to be long-lived, potentially living over 200 years. Its diet, documented from stomach contents, includes fish, squid, octopus, and marine mammal carrion. A Pacific sleeper shark was once filmed eating a sperm whale carcass at depth off Hawaii, one of the few direct observations of a large deep-sea shark feeding in situ. Most of what is known about the diet and ecology of Pacific sleeper sharks comes from incidental capture in fishing gear and from the examination of stomach contents, not from direct observation. The deep-sea rays, belonging to several families of skate and ray, are similarly poorly characterized in terms of their living biology. They are often the largest invertebrate predators on the continental slopes, feeding on fish and large invertebrates. New species of deep-sea skate have been described in recent years from specimens collected in previously unsurveyed portions of the Pacific and Indian Ocean slopes. The patchwork nature of deep-sea knowledge, very detailed in some areas and almost blank in others, reflects the history of which geographic areas have had research ships visiting them and which have not. The North Atlantic, the Eastern Pacific, and the waters around Australia and New Zealand are among the better surveyed deep-sea regions. The deep-sea slopes of the South Atlantic, much of the Indian Ocean, the Western Pacific outside Japan, and the waters around Antarctica are less well surveyed. The distribution of research effort mirrors the distribution of wealthy nations with active marine science programs, which does not match the distribution of biodiversity in the deep ocean. This geographic bias in sampling has implications for the total estimate of deep-sea biodiversity. If the areas sampled have been the areas near large marine science programs, and those areas have particular characteristics, then the species found there are not representative of the global deep-sea fauna. Extrapolating from a biased sample to a global estimate produces estimates that are systematically biased in ways that are difficult to correct without more comprehensive sampling. The development of autonomous deep-sea instruments, including autonomous underwater vehicles that can operate at full ocean depth, has the potential to address some of this geographic bias by reducing the dependence on expensive ship time. Several national and international programs are developing autonomous systems capable of collecting biological and oceanographic data from the deep ocean at geographic scales and temporal resolutions that ship-based surveys cannot match. These systems are becoming operational progressively, and the data they generate will substantially change the picture of deep-sea biodiversity over the coming decades. The environmental DNA approach, already mentioned in the context of the mesopelagic zone, is applicable throughout the water column and into the sediment. A liter of deep-sea water contains genetic material shed by every organism that passed through or lived in that water recently. Filtering and sequencing that material produces a list of genetic sequences that can be matched against databases of known species or flagged as unknown. The technique has been applied to deep-sea water samples and sediment samples from multiple ocean basins, consistently detecting a substantial fraction of sequences that do not match any described species. The interpretation of environmental DNA data is complicated by several factors. The genetic material in a water sample may have come from organisms present at the time of sampling or from organisms that passed through hours or days before. The geographic origin of the material is uncertain because ocean currents transport shed DNA. The taxonomic assignment of unknown sequences depends on the completeness of the reference database, which for deep-sea organisms is far less complete than for terrestrial groups. Despite these complications, environmental DNA is providing a view of deep-sea biodiversity that is broader in geographic scope than any previous method, and it is consistently showing that the number of distinct biological entities in the deep ocean is larger than the number of described species. There is one more aspect of the unnamed deep-sea world worth discussing before the documentary draws to its natural close. That is the question of what the unnamed things mean for the ecosystems that contain them. Ecosystems function through the interactions between their components. Who eats whom, who decomposes what, who transforms which chemical compounds, who provides shelter for whom. When a component of an ecosystem is unnamed, it is not necessarily unknown in ecological terms. It may be present in trawl samples. It may appear in stomach contents. It may be detected acoustically. It may leave chemical traces in the sediment. But without a name, it cannot be consistently tracked across samples from different locations or different times. Without a name, the scientific literature on it is scattered and difficult to compile. Without a name, it is difficult to assess whether its population is stable, changing, or responding to environmental pressures. The unnamed organisms of the deep ocean are doing ecological work. They are eating, being eaten, decomposing organic material, fixing nutrients, carrying carbon from one depth to another, providing attachment surfaces for other organisms, sequestering chemical elements in their bodies. The sum of this work is enormous, at the scale of the global ocean. Some of it, particularly the role of mesopelagic organisms in the biological carbon pump, has been estimated to have significance for the global carbon budget comparable to the role of terrestrial forests in the global carbon cycle. Those estimates carry large uncertainties, partly because the organisms doing the work are poorly characterized. The deep ocean is, in this sense, not just biologically unknown but geochemically significant in ways we cannot fully quantify because we do not know the participants well enough. The scale of what we are missing in our understanding of the deep ocean is not just a matter of scientific completeness. It is relevant to understanding how the ocean functions as a system, how it responds to change, and what role it plays in the larger chemistry of the planet. All of these things are happening tonight, in the dark, in the cold, under pressure, without any requirement that anyone know about them. The ocean is full of unnamed things. They are doing fine. The water above them is dark and old and very large, and that, for tonight, is enough. There is a specific fish worth ending on before the documentary truly closes, not because it is the most dramatic or the most alien of the deep-sea organisms, but because it demonstrates the ongoing nature of discovery in the most immediate terms. In 2019, researchers from Scripps Institution of Oceanography, working with a remotely operated vehicle in deep water off the coast of California, filmed a fish at approximately 1,000 meters depth that they could not immediately identify. The fish was roughly half a meter long, eel-like in body form, with large eyes and a distinctive color pattern. The footage was analyzed by fish taxonomists, who compared the appearance with every known deep-sea fish species from the eastern Pacific. No match was found. The footage was shared more widely within the deep-sea fish community. Still, no confident identification emerged. The fish was subsequently given a working designation as a potentially new species, pending the collection of a physical specimen for formal morphological and genetic analysis. Whether it is truly a new species or an unusual variant of a known species has not been definitively established. The footage exists. The fish exists in the footage. Its identity is unresolved. This kind of event, a filmed but unidentified organism from a relatively accessible depth range in a well-studied ocean basin, is not rare. It happens on a significant fraction of deep-sea ROV dives. The database maintained by MBARI of all biological observations from their ROV dives contains a category for unidentified organisms, and that category is not small. The unidentified observations include organisms that taxonomists later matched to known species, organisms that were provisionally identified but not confirmed, and organisms that remain genuinely unidentified after consultation with specialists. The Monterey Bay Aquarium Research Institute has been conducting deep-sea research since 1987, with a focus on Monterey Canyon and the adjacent deep-sea environment. This is one of the most intensively studied deep-sea locations in the world. And even in this well-studied location, after decades of ROV operations covering the water column from the surface to over 4,000 meters depth, new biological observations are made on a regular basis. The deep sea, even in places that have been consistently surveyed for decades, continues to produce things that were not there before, or that were there before but were not seen, or that were there before and were seen but not recognized. The Humboldt squid, Dosidicus gigas, is one of the better studied mesopelagic predators because it is commercially fished in large quantities in the eastern Pacific and because it can be maintained briefly in captivity for behavioral studies. It can reach 2 meters in length and over 50 kilograms in weight. It migrates vertically, spending daylight hours at depths from 200 to 700 meters and ascending toward the surface at night to feed on fish and smaller squid. It is highly aggressive, equipped with toothed suckers on its tentacles that can lacerate prey and the skin of divers who have had close encounters with them. Despite being commercially important and relatively well studied, the details of Humboldt squid reproduction remain incompletely understood. The location and nature of their spawning aggregations has not been directly observed. The development of eggs and larvae has been partially documented from specimens, but the early life history in the open ocean is poorly known. Their communication, which appears to involve rapid color changes across their chromatophore-bearing skin, has been studied in brief encounters, but the content of the communication, whether these color changes convey specific information between individuals, is not established. The bioluminescent patterns on Humboldt squid skin have been recorded and analyzed. The patterns appear to change in response to social context, with different patterns observed when squid are alone versus when they are in a group, versus when they are feeding. But the interpretation of these patterns, what they mean to another squid, what behavioral response they produce, is not known. The experiment that would answer this question, presenting a squid with recorded bioluminescent patterns from another squid and measuring the behavioral response, has been technically difficult to execute because the high-speed chromatophore changes are hard to reproduce artificially. This difficulty in studying the communication systems of deep-sea and mesopelagic cephalopods is representative of a broader methodological challenge. Many of the most interesting questions about deep-sea biology are behavioral questions, and behavioral research requires either live animals in their natural environment, which is difficult, or live animals in laboratory conditions that accurately replicate the natural environment, which is also difficult for deep-water species. The gap between what we know about the anatomy and biochemistry of deep-sea organisms and what we know about their behavior is large and is unlikely to close quickly. The basket stars, belonging to the order Euryalida, are echinoderms that look nothing like the typical starfish image. They have 5 primary arms that branch repeatedly into finer and finer ramifications, producing a structure resembling a branching tree or a complex net. The basket star uses this structure to collect food from the water column. It extends its arms into the current and traps small organisms and organic particles in the branches. When disturbed or exposed to light, it contracts all its arms into a compact ball. The largest basket stars, found in deep water, can reach arm spans of nearly 1 meter when fully extended. Basket stars are found in deep water across all ocean basins, often on the tops of seamounts or along the ridges of mid-ocean ridge systems, where current flow is strong and provides a continuous supply of suspended particles. They are relatively long-lived and slow to reach reproductive maturity. Their population ecology is essentially unstudied for most species. The total number of basket star species is not confidently known. The deep-sea medusae, the jellyfish of the deep ocean, include some of the most visually striking organisms ever filmed by ROV cameras. Atolla weevillei, a common deep-sea jellyfish of the bathypelagic zone, has a bright red bell with a long trailing tentacle and a crown of shorter marginal tentacles. When disturbed or attacked, Atolla produces a bioluminescent display called the burglar alarm, a rapid, rotating wheel of blue light around the bell, which appears to attract larger predators to the location of whatever is attacking the jellyfish. If a small predator is eating the jellyfish, the light display attracts a larger predator that then attacks the smaller one, allowing the jellyfish to escape. This is bioluminescence used defensively, not to dazzle the predator directly, but to summon a larger threat to the location. The burglar alarm behavior has been confirmed experimentally and is now considered one of the more elegant examples of indirect defense in marine biology. But Atolla weevillei is a relatively well-studied deep-sea medusa by the standards of the group. Most deep-sea jellyfish are known from occasional encounters, fragmentary specimens, or footage in which the animal was observed but not collected. The diversity of deep-sea medusae at abyssal and bathypelagic depths is not confidently characterized. New species are described sporadically from the relatively rare occasions when an ROV encounters a jellyfish and either collects a sample or captures footage good enough for detailed analysis. The midnight zone, a popular term for the bathypelagic zone from 1,000 to 4,000 meters, earns its name from the fact that no sunlight of any wavelength or intensity penetrates to these depths under any conditions. The only light in the midnight zone is bioluminescence produced by the organisms themselves. And yet, the midnight zone is not simple or uniform in its biological content. The organisms there occupy niches as specific as those of any surface ecosystem. There are filter feeders, active predators, scavengers, parasites, detritivores, and primary producers in the form of chemoautotrophic bacteria. The ecological architecture of the midnight zone is broadly similar to that of a surface ecosystem. The specific organisms filling each role are different. The fang-toothed fish, Anoplagaster cornuta, is among the more dramatic-looking of the midnight zone residents. Its teeth, proportional to its body size, are the largest of any fish. The longest teeth are so large that the fish cannot fully close its mouth. The teeth slide into sockets beside the brain when the mouth is at rest. The fangtooth is a predator, using these teeth to grip fish and squid caught in the dark water. Its body is compressed and dark, and it is found throughout the bathypelagic zone in tropical and subtropical waters globally. Despite its striking appearance, the fangtooth is not well studied. Its abundance in different ocean regions is not known. Its reproductive biology is not documented in detail. The only direct observations of fangtooth behavior in the wild have been incidental encounters during ROV dives, in which the fish typically retreats rapidly from the lights of the vehicle. What it does in the dark, how it locates prey, how it interacts with other individuals of its species, is not known. And so it goes, through the long catalog of deep-sea organisms. The well-named and poorly known. The partially known and partially named. The detected but unseen. The seen but unidentified. The collected but undescribed. The ancient and the unchanged. The new and the surprising. The enormous and the microscopic. The organisms doing work that matters to the chemistry of the planet, without any record of their existence in any scientific database. The deep ocean is not going anywhere. It is as large tonight as it was last night and will be tomorrow. Its contents are not waiting for us to notice them. They are not diminished by our ignorance of them, and would not be enlarged by our knowledge. They simply are, in the dark, in the cold, at pressure, doing the things that the conditions of their existence have shaped them to do, on timescales that make any human observation a brief and local thing. That is where we leave it for tonight, not at an endpoint, because this is not the kind of subject that has one, at a resting place, in the middle of something very large. with more in every direction than any inventory has captured. The ocean keeps its own count. It has always been larger than our count of it, and it will still be larger tomorrow. Consider what it means for something to be unknown, not to us individually, but to science as a collective enterprise. The scientific literature contains, at present, formal descriptions of approximately 250,000 marine species. Each description is the result of someone looking carefully at an organism, comparing it with existing descriptions, measuring and photographing it, writing a detailed account, and submitting that account to a peer-reviewed journal where other specialists confirmed that the organism had not previously been described. That process, when it works correctly, produces a permanent record. This organism exists, it has these characteristics, it was found in this place, and its name is this. The organisms that science has not yet described are not less real for the absence of that record. They are present in the ocean, interacting with other organisms, playing their ecological roles, occupying their ecological niches. They have bodies, metabolisms, behaviors. They are alive in exactly the same sense that the described species are alive. The description, the name, the entry in the database are human conventions. They matter for human science. They do not matter to the organism. This is something worth holding on to during the long, quiet hours of the night. The ocean is full of living things that have no name in any language. They were alive before language existed. They will be alive in places where language has never reached. The naming is something we do for ourselves, to be able to talk about what we find. The things themselves preceded the talking and will outlast it. The deep-sea holothurian moving across the abyssal sediment at 4,000 meters depth does not require a scientific name to move. The Magnapinna squid holding its extraordinary arms at right angles in the bathypelagic zone does not require a formal description to exist. The lanternfish rising toward the surface in the dark hours of every night across every ocean on Earth does not require our count of its species or our estimate of its biomass to conduct its migration. The ocean does what the ocean does. It has been doing it for a very long time. We are relatively new to paying attention. We are paying more careful attention now than we were a century ago. We will pay more careful attention in the future than we do today. The picture will become more complete. The names will accumulate. The descriptions will expand. The ecological relationships will become clearer. The role of the deep ocean in the chemistry and physics of the planet will be better understood. And there will still be things in the deep ocean that have not been seen. There will still be sounds in the SOFAR channel that do not match any known source. There will still be genetic sequences in the environmental DNA databases that do not match any described species. There will still be footage from ROV dives that shows something moving in a way that no specialist can immediately explain. There will still be organisms in museum jars from expeditions decades ago, preserved and labeled with a date and a location, waiting for someone with the time and expertise to look at them properly. The deep ocean is not a problem that will be solved. It is a place that will be known, progressively, by degrees, over a very long time, by people who choose to direct attention toward it. Tonight is one moment in that long process. The deep scattering layer is rising in every ocean as you listen to this. The lanternfish are ascending, the hatchetfish are following. The bristlemouths, the siphonophores, the gelatinous zooplankton, all of them moving upward through the dark water, through the pressure gradient, toward the surface where the food is tonight. Below them, the abyssal plain extends in every direction. The xenophyophores are in the sediment. The sea pigs are moving slowly across it. The ossidax worms are in their bones. The polychaetes are in the sediment between everything else. Deeper still, the hadal fauna of the Mariana Trench are going about their business at pressures that would destroy any material we put there without extraordinary engineering to protect it. And somewhere in the Southern Ocean, in water that is cold enough to be at the edge of freezing, in depths between 1,000 and 2,000 meters, a colossal squid is alive. No camera has ever captured it. No researcher has ever seen it. Its existence is documented only through fragments and the stomach contents of sperm whales. But it is there. It is the largest invertebrate on Earth. It has been there in that form, long enough for the lineage to leave traces in the fossil record. And it is not waiting for anyone to come and find it. It is simply there, in the cold, in the dark, doing what it does. The ocean is very large tonight. It was very large last night. It will be very large tomorrow. And that is not a mystery waiting to be solved. It is a fact to sleep beside. There is no final inventory of the deep ocean. There is only the ongoing work of people looking carefully at a place that is very large and very old and full of things. The names accumulate slowly. The discoveries keep coming. The ocean holds more than any count has found. It held more before anyone counted. It will hold more after the counting stops. What is really living in the deep ocean that science has not named is not a mystery in the sense of a riddle waiting for a solution. It is a description of the current state of human knowledge about a place that exceeds that knowledge by an enormous margin. It is not a reproach. It is simply the situation. And the situation is one in which there is and will remain for a very long time more in the ocean than anyone has named. Tonight, that is enough to know. The rest is still there, still unnamed, still doing fine.